second day by the pharmaceutics session. And the first lecture will be held by Dr. Abdelkader Metwalli. Dr. Abdelkader got his master in 2006 from Ain Shams University in Egypt. In 2012, he received his PhD from Bath University in UK. Then he returned back to Ain Shams University, where he is currently a professor. Starting from 2018, he joined Kuwait University in the Department of Pharmaceutics. Nanomedicine and computational pharmaceutics are his main areas of interest. And with these areas, he co-authored 28 papers. Today, he's gonna give us a lecture on artificial intelligence and the role of machine learning in drug delivery. So join me, please, to welcome Professor Abdelkader.
Thank you, Dr. Noah. Good morning, everyone. Thank you for coming to this presentation. Artificial intelligence is changing the world around us. We use it every day in face recognition applications, with ChatGPT language models, when we do a web search, and indeed, it is having an increasing importance in pharmaceutical research. So the aim of this presentation is to provide the information about the application of AI in scientific research, to be familiar with the terminology and workflow of AI applications, and to provide examples of the use of AI in pharmaceutical research. We will start with a brief introduction to AI, and then we will shift our focus on machine learning and its workflow as a type of AI applications. Then we will provide few examples of pharmaceutical applications of machine learning, and finally, we will discuss with a bit of details a pharmaceutics example, predicting the in vivo efficacy of small interfering RNA lipid nanoparticles. So what is AI? AI is a general term. It refers to the development of computer systems that can perform tasks that requires human intelligence. And this is like a broad field. So it contains many disciplines and tools, among them machine learning, where the computers learns from data. It includes natural language processing, like ChatGPT is a common example in robotics. However, we will focus on machine learning as it is widely employed in pharmaceutical research and other disciplines. So machine learning by itself, it focuses on analyzing and interpreting patterns in data. It detects hidden patterns, structures in the data. And then accordingly, it does something with this data, make a prediction, make a decision, change the organization of the data, but it involves examining the data. The learning aspect itself can be defined as a process by which the computer improves its performance with respect to a specific task. So this means by a repetitive examination of the data, the hidden patterns and trends within the data, the computer can improve its performance, for example, can improve its predictions of a certain outcome. And again, the central idea here is observing and analyzing the data. So what is the difference really between machine learning as a type of AI and classical programming? In classical programming, we provide the computer with instructions. So for example, we instructed the computer to do the following. If the patient's weight is this, if the patient's total blood cholesterol level is that, if the patient is 40 years old, so set of conditions. Then we instruct the computer to do some action. For example, select a specific drug that is suitable for this patient. So all the way, we are providing instructions to the computer, conditions, and what to do about them. This is the conventional or classical programming. However, the machine learning way, it involves first providing the computer with data. And the computer then examines this data. Data like, for example, include patients, like these are our observations, their characteristics, their age, their weight, their blood sugar level, and so on. Maybe some therapeutic outcome, their blood pressure. This is the data that the computer examines. And then, according to what the computer finds on its own, we don't interfere with this. We don't tell the computer if the patient's age is this, if the patient's weight is that. We don't interfere with this. The computer detects these patterns within the data. The computer then, after it learns, it makes some decision. Again, makes some prediction, select a specific drug, and so on. So here, in the machine learning way, the computer learns from the data without human interference. The only human interference here is guiding the learning process itself, as we shall see in the following slides. But there is no human interference with respect to the patterns themselves, what is detected from examining the data by the computer, or the following decisions by the computer regarding this data. So this is the main difference between the classical programming and machine learning. So as such, machine learning can be categorized into two main groups, the so-called supervised learning and unsupervised learning. Supervised learning is when we have some independent variables, features, and a dependent variable, an outcome, a response. So for example, a feature is the patient's weight. A response would be its blood pressure, and we want to predict the blood pressure based on some characteristics of this patient. This is called supervised learning. There are inputs, and there is a response, or maybe even more than one response. And this even can be categorized into classification tasks if we have categorical responses, like treated or non-treated, high, intermediate, low levels of some interesting factor, or regression tasks where we have numerical or continuous data. The unsupervised learning, on the other side, we don't have a response, but we usually investigate the structure of the data that we have. So it comes to mind the clustering, grouping, for example, formulations that are similar together, or patients with similar characteristics together. Dimensionality reduction, if you want to decrease the number of the features, the number of the properties of the observations for better visualization or for modeling later on. This is called unsupervised learning. So it is very common in the literature to find these methods. So for example, in the supervised learning, we might find the use of artificial neural networks, random forest Gaussian processes, support vector machines, random forest gradient boosting, and so on. All of these are machine learning methods used for classification and regression tasks, used for supervised learning. While in the unsupervised learning, there are techniques that are commonly used. Among the most famous of them, maybe the principal component analysis, the hierarchical cluster analysis, and such techniques that are related to the structure of the data without predicting any outcomes or responses. It is worth mentioning here about the different types of supervised learning, that all of them has their strengths and their weaknesses, actually. So for example, regarding the computational time taken by each of these to construct the predictive models, some of these methods are sensitive, takes longer, much longer times, with an increase in the number of observations. We are talking about the number of observations in a data set might be tens or hundreds or thousands, or tens or hundreds of thousands, or even millions. So some of these methods are sensitive to the number of observations. Others are sensitive to the number of features, the properties. Again, this number of features can be anything from few of them up to thousands of them. Some are sensitive to both. Artificial neural networks and deep learning are sensitive, for example, to very large data sets. It takes a lot of time to construct the predictive models. Another difference is whether the method can be explained, or the result, the model can be explained or not. It depends really on the aim of the research project, or what do we want to do with machine learning here. Because some methods, like, again, the artificial neural networks, are called black box methods. They provide excellent predictions, but the models are very difficult to explain. Other methods provide information and can be explained. The models can be explained, so all tree-based methods like the random forest or extreme gradient boosting methods, they provide information about the importance of the variables that are used in the model. So if we want to do this, usually one or more of these are chosen. Another difference is that actually all of these, machine learning in general, is data-driven, which means that there is no specific method is better than the other. It all depends on the data set and the aim of the research project. So for one project, neural networks might outperform the support vector machines. In another project, support vector machines might outperform the artificial neural networks. So these are among the factors that governs the choice, why some papers use random forest, why some papers use artificial neural networks, and so on. So how this is done, generally speaking, because there is a general or universal workflow for using machine learning in scientific research, and indeed in the different disciplines of pharmaceutical research. So there is this machine learning pipeline. First, we must have the data, because the computer learns from the data. Then we must select the suitable machine learning method. And finally, a very important step is the validation or the evaluation of the models. So regarding the data, we must take care from where do we obtain this data, its preparation, its inclusion, exclusion criteria. More on this will be when we discuss in details the pharmaceutics example. For example, we can get data from the published literature, data mining. We can obtain data from our own labs. It is lab generated. Maybe we do some chemoinformatics or bioinformatics work to generate molecular descriptors or so for the structures that we are working on. So we must take care of the sources of the data. And then we must, according to the objective of the research, the type of learning, we choose the appropriate method of machine or methods of machine learning, like for regression, let us say artificial neural networks, random forest, and so on, again, depending on the size and properties of the data set and the aim of the research project. And finally, and very important, the evaluation of the models, because usually, regardless of how this is going to be done in a specific research project, usually there is a group of observations or samples, patients, for example, or formulations that are kept aside. These are not involved in constructing the predictive models. They are kept aside to the end to test the predictive performance of the models that we have on these held out samples. Usually the samples or observations that are used to construct a predictive model, these are called the training set, and the samples that are kept aside to the end to predict the outcome in unknown samples at the end, these are called the test or validation set, or held out group. It really depends on how this is done, but the idea is the same. Some of the observations are kept aside to the end to validate the predictive performance of the models. And there are many metrics that are used to quantify this generalization capability, predicting unknown samples, samples that have never been used before in constructing the model. So for regression tasks, the root mean squared error, the correlation coefficient, or coefficient of determination, the R squared, the mean fold error, or for classification tasks, accuracy, specificity. Really, again, it depends on the aim of the project and what we want to do or conclude from this project. So these steps will be repeated almost in every single machine learning project or scientific research involving machine learning. So let us see a few applications of this in our own research in the pharmaceutical disciplines. To start with, if we like to show how the increased use of machine learning in research, in pharmaceutical research, a simple search on PubMed shows this. These are the years from 2005 to 2024, and the trend is the increased use of machine learning in research related to pharmacy or pharmaceutical sciences. So we have more than 2,500 publications. This was obtained like a few days ago. So indeed, the trend is the increased use of machine learning, and for good reasons, as we shall see. So we will start with some applications of AI in pharmacy practice, paper by Yeltsin et al. The aim of this project was to develop a model that predicts the presence of medication errors in neonatal ICU unit. So to do this, they must have the data to begin with. This is what the computer is going to learn from. So the data was obtained from prescriptions of 412 patients. Some medication errors were found in some of these patients' prescriptions, or whether a medication error during preparation or in the prescription or for the administration. The parameters of features related to this, some of them were related to the drugs, so the number of drugs used, for example, in a specific prescription. Some of them was related to the healthcare professionals' working hours, total working hours. Some of them was related to the patients themselves, the postnatal age, and so on. So this is how they prepared their data, and this is a very crucial and important step in any machine learning project. Then they split the data to a training set. This is what the models are going to be constructed from, the train set, and a test set, which is kept aside to the end to test the predictive performance of this model on unknown samples or observations that the model has never seen before. The machine learning model here, the task itself, it was a classification task because it is the presence or absence of a medication error. And they used one technique called random forest to do so. The validation or evaluation of the model resulted really in excellent performance. This is 92% success in predicting the presence or absence of medication errors. So we can see that this is of help in any healthcare setting where by using this model, the computer can point out actually to the prescriptions where a problem might be expected. So the pharmacist or the healthcare professional can take care of this. So this is one example of application in pharmacy practice. Another example that can be used in pharmaceutical chemistry is the prediction of protein ligand binding affinity. And this is, of course, to select the best candidates to further develop these drugs. And the premises here by the authors was that once the models are constructed, it is very fast to make these predictions. So here there is a difference between constructing the models, doing the machine learning work itself. It takes a lot of time, actually, and a lot of computational time. So we might be talking about weeks, months, or more, years. It depends on the project. But once the predictive models are constructed, this is usually very fast to make the predictions as compared to the other wet lab, of course, or even computational methods used in the context of pharmaceutical chemistry. So as compared to the molecular dynamics simulations, or as compared to docking and docking positions refinement in the pharmaceutical chemistry, making predictions with machine learning should be much faster, and this is the advantage. So again, what they have done, they have prepared their data. They obtained the information from PDB bind database. They had this number of observations, 2,800 plus, and 128 features in columns. The rows represent the protein ligand pair, and the columns are their properties. The features, some of them were specific to the ligands themselves, when we talk about features and descriptors of this type, it's the types of atom counts, log P, topological surface area, maybe number of hydrogen bonds donor, number of hydrogen bonds acceptors, and there are thousands of these chemoinformatic descriptors. And some of the features were specific to the protein itself, like the number of SS bridges and the number of chains. And this is a regression task since the binding affinity, this is a numerical continuous variable. Again, they selected the random forest and the Gaussian process to do the task. And here in this research, the random forests outperformed the Gaussian process and resulted in an R squared and root mean squared error of 0.76 and 1.31 respectively. And again, the advantage of this is that once this work is done and the predictive models are prepared, making predictions with them is very fast compared to other methods. And of course, as long as the required accuracy is achieved. Another example in the pharmacology or toxicology setting was the risk assessment of drug-induced liver injury. So again, the preparation of the data, these were data of 96 compounds obtained from the liver toxicity knowledge base. The features were diverse from in vitro assays using mitochondrial toxicity and such assays, and other physicochemical descriptors regarding the compounds like log P or even pharmacokinetic, we can call them descriptors like Cmax and similar pharmacokinetic parameters. And the model itself was a classification task model. This is classification. Again, they are going to classify the compounds as having no or low toxicity, or medium or high toxicity, or can induce liver injury, highly likely to induce liver injury. And the thing here, when they chose this type, the Bayesian model, other than doing the classification task, it is very useful in providing probability that actually this prediction is accurate, that actually this specific compounds falls within this specific class, which is a benefit, of course, to know if a predicted value is reliable or not. And again, the validation of this model was carried out with accuracy of 63% on held out samples, the samples that was not used to construct the model. And for a binary yes/no liver injury prediction, a balanced accuracy of 86%, sensitivity of 87%. These are metrics used for classification tasks. So again, this is of great benefit when predicting beforehand whether a drug is a good candidate or it will cause certain side effects or not, and so on. So how about the pharmaceutics? And we will discuss with a bit of detail here an example. In pharmaceutics, we can do many things with machine learning, really. So we can predict drug loading in nanoparticles. We can predict drug release at certain time points. We can predict some pharmacokinetic parameter. Maybe we can predict change in solubility by formulating the drug in maybe micelles or so. Predict in vivo performance, like the example that we are going to discuss. So this is a work that I participated in. I was a co-author here. In silico prediction of siRNA ionizable lipid nanoparticles in vivo efficacy, machine learning modeling based on formulation and molecular descriptors. This was published in 2022. So the idea here, small interfering RNA is relatively large molecule compared to small molecule drugs. It has a lot of negative charges on it because of the phosphate backbone. So it is difficult for it to get into the cell as such. So it requires a carrier to deliver it inside the cells. The carrier neutralizes the positive charges and at the same time protects the small interfering RNA from degradation in the biological fluids by the nucleases present. So one such a carrier system is lipid nanoparticles. And this is a system that is similar, for example, to the Pfizer messenger RNA vaccine, the same idea. So here, this is the double-stranded small interfering RNA. Usually in the formulation, there are cationic lipids. This is a scheme of that shown in red here to neutralize the negative charge and complex the small interfering RNA by electrostatic interactions. There are other components, co-lipids or helper lipids, that either facilitate the release of the cargo or making the drug delivery system responsive to external stimulation like change in pH or enhancing the stability of the drug delivery system. For example, cholesterol is commonly used for such purposes. And also, usually these systems have some sort of PEG, polyethylene glycol coating, to prevent the rapid clearance and rapid removal of the nanoparticles from the blood, to protect them from opsonization and protect them from being uptaken by the RAS system and by the macrophages. So the wet lab approach to do this is to design the cationic lipids, synthesize the cationic lipids, purify them, characterize them, and so on, and then formulation of the nanoparticles themselves, selection of the excipients, finding out the optimal percentage of these excipients, and then evaluation of these and characterization of these nanoparticles in terms of their size, physicochemical properties, their stability in the biological fluids, and then their performance in vitro in the different or relevant cell cultures, and in vivo. And all of this is preclinical. However, as we can see here, this is time and resources consuming. So the question here, can something be done here about this? So the objective of the paper or work that we are going to discuss was that given that we have formulation variables, for example, PEG mole percent on the surface of the nanoparticles, structures of the cationic lipids, a required knockdown percent. The siRNA, as we know, it acts by knocking down genes. They decrease or maybe completely inhibits the expression of a specific gene. So for a specific gene, there is a specific siRNA that will cause degradation of the messenger RNA of this gene. Thus, it decreases or maybe almost completely inhibits its expression. So given all of that, could the in vivo siRNA dose be predicted for a specific formulation or not? And the thing here, the siRNA dose is very important and is taken as a parameter for the efficacy or potency, depending on the context of the siRNA lipid nanoparticles, because siRNA on its own has its own issues. First of all, there are off-target effects. If there is any or there are any complementary sequences in any other genes, this might cause the knockdown or downregulation of these unintended genes. So we have off-target effects. There are immunological effects. The sRNAs can, for example, interact with the toll-like receptors and cause a cascade of events. So this is unwanted, of course. Also, they are costly. To prepare small interfering RNA for human use, it is usually very costly. So decreasing the dose is a good sign of efficacy and safety of the formulations. And also putting in mind that lower dose and lower dose of the nanoparticles means lower amount of the cationic lipid delivered, and usually also cationic lipids have their own share of toxicity because of the way they interact with the different cellular components, because of the positive charge. So could the in vivo Serna dose be predicted for a specific formulation or not? And the way we approached this was as follows. First, the sources of the data. We obtained the data from 5 publications by 5 different research groups. It included the cationic lipid structure, because the structure of the cationic lipid makes a difference. They are not equivalent. Cationic lipids, being cationic only, is not the only factor. Their structure affects their efficacy and their safety. How would they efficiently complex the sRNA, and how they are going to release it inside the cells, and also any associated toxicity with these cationic lipids. The certain formulation parameters like the PEG%, because it has an effect on the activity actually. The small interfering RNA dose, in these publications, the sRNA was against factor 7 gene in mice, and the in vivo knockdown percent of these small interfering RNA delivered by these lipid nanoparticles. And it is very important here to prepare the data set for the following machine learning properly. So what is usually done, and what we have done here, first of all, there must be specific inclusion criteria. Which publications are going to be selected in the first place, and what should be there? So, for example, we should have all of the required information there. Sometimes the reporting of the amounts of excipients is not clear, so we have to take care of that, if it can be calculated or not from the information present in these publications. Then, following this, we generated some 1D and 2D descriptors using chemoinformatics tools, depending on the structures of the cationic lipids. We curated the data set curation, an important step. So we removed any entries with NAs or missing values or any repetitions or any highly correlated features, so features that have a correlation coefficient of, let us say, 0.99, they are highly correlated, so essentially they are the same. And they will be problematic, they will increase the complexity of the models, they might lead to poor predictions later on. So they were removed also to make the size of the data set smaller, so it is faster to prepare, construct the predictive models. And then we carried out data splitting into training and validation partitions, training for construction of the models, and validation for evaluation of the prediction capability of the prepared models. This was followed by the machine learning itself, and here it was actually 2 parts. One part was the selection of best features by an evolutionary algorithm. This step is carried out usually in machine learning research to, as it is written, to select the best features or properties that can be used in the model, because other irrelevant properties, usually, if they are present, will lead to poor predictions. So we need to select only the useful predictors or features or properties. So in the literature, if you read any literature, regardless of the discipline, this step you will find this terminology. You will find either feature selection or feature extraction, sometimes feature engineering when we create new features from the original ones, but they all aim exclusively to the same thing. We want to select the best features for the model and exclude the non-useful features. So this was done, and simultaneously, supervised learning was carried out. This is a regression task, so we used 3 algorithms: the artificial neural networks, support vector machines, support vector regression, and partial least squares. And finally, the model was validated on the validation by evaluating the predictions of the validation sets, those samples that were never used in the feature selection or in the supervised learning tasks were held out to the end, and also by a technique called Y randomization of responses. This technique simply tests if the original models were due to chance or not. So what we do here is that we we randomize the responses, the doses of the siRNA. So instead of formulation 1 having this dose of siRNA, formulation 1 will have the siRNA dose of the formulation number 100, and formulation 2 will have the dose of formulation number 10, and so on. We scramble this, and the idea, if the predictions after these randomizations are much worse than the predictions of the original models, then this means that the original models are actually true models, and the relationships there are meaningful. They are not due to random chance. Another thing to point out here, that the part of the machine learning itself in this work was carried out by the R programming language using an R software, and it is very useful, really. The R software, it is free, it is open source, and it has a wide, really set of machine learning methods and visualization packages, functions, they call them packages or libraries that can be called. We can think about this as an add-on, like an add-on in any of the applications that we use. So we can call these packages with the functions or methods that we want to apply in our own program. So we used the R language, and the thing is, the R requires scripting, really. So we write the program itself, how the learning will be carried out. So this is a snapshot of one of the scripts that we are using at the moment in a project. These scripts can be short, it can be relatively long. So if we print it, for example, on an A4 paper, it can take 3 or 4 pages. But really, once the basics of writing these scripts is learned, everything comes fast afterwards. It's not like difficult or anything. And again, the advantage of this, that it makes constructing the models and adjusting the parameters of the models a very flexible process and under control. We can control everything from A to Z in the learning process. Another alternative also to the R package would be the Python environment using the Python programming language. Although it is my personal opinion that I prefer R, it has more methods and more packages, and the list is growing, really, that we can use. So how this worked out? First of all, the data set that was obtained, we obtained a data set with 120 entries, formulations. The features were 438 features after processing the data set, and the response was the log siRNA dose. So here, first, we started to examine the structure of the data just to make sure that the validation observations are uniformly distributed among the training observations. So here we did this splitting of the 120 entries into a training set that has 90 of them, and 30 were kept aside for later on to validate the models. And because really when we select from these 120, select 90, we have numerous ways of doing this, and like almost endless probabilities of which exact entries are going, which 90 of the 120 is going to be selected. So we initially started with 2 different splits or 2 different sets. So each of this is one set and this is another set. Here we have 90 observations for training, and here also we have 90 observations of training. The difference is which observations are included. For example, here might be formulation number 1, 2, 3, and here maybe 1, 2, 5, and so on. So they differ in their composition. But at the end of the day, each of them has 90 training entries and 30 validation entries. And the colored triangles here, whether in the pink color or the orange color, represent the validation entries. The carrying out principal component analysis and plotting the scores of the PCA against each other, the principal components, the principal component 1 against 2 or 1 against 3, these are the components that hold most of the information in the data set. So they are very useful in the visualization of the data set. And what we want to make sure of is that the validation observations are uniformly distributed almost in the same manner as the training observations. So this is important to prepare good predictive models and important for obtaining reliable validation of the models when we predict the responses of the validation entries. Then the machine learning was carried out using 3 different methods. We have 2 sets. So this is on the left-hand side, set 1. On the right-hand side, we have set 2. And in the first row, this is the artificial neural networks. The second is the support vectors regression. And the final one, the lower one, is the partial least squares regression. And we can notice many important things here. First of all, that they don't result actually in the same quality of predictions. There are differences. So because they are data-driven models, even the same artificial neural network, the same method, but on using different training sets and different validation sets, there is slight difference in the performance because, again, they are data-driven models. And there are differences also between each method of machine learning. And the important thing here, we considered, or we concluded that the artificial neural networks, the top row here, outperformed the other 2 methods because we were mainly interested in the predictions on the lower side. Here is a plot of the actual log of the dose against predicted log of the sRNA dose. And we were interested here with the accuracy of the predictions on the lower side because we are interested in doses that actual doses are like 0.0 something, 0.01, 0.03, because again, these are the doses that are going to avoid the off-target effects and the side effects of the small interfering RNA and of the cationic lipids at the same time. So it is important to obtain these accurately as opposed to the predictions on the higher side of the doses. So for example, if the actual dose is 1 or 1 point something, this is a very high dose and might actually lead to side effects. So regardless of the error here, they are all bad, technically speaking. But here it is important to get them right because these are the good candidates down here. And on the lower side, we notice clear differences between the different methods. Of course, the artificial neural networks got the predictions here right. When the predictions are close to this red line, this means they are exactly like the actual value. But as they go away from this line, they get more far away from this line. This means that there is an error, and the error increases as the distance increases from this red line. And these are the metrics of the figures that we have just seen. So for the artificial neural networks, whether in set 1 or in set 2, we notice that the root mean squared error is 0.23 or 0.3. So the lower the value of the root mean square error, the better. So for set 1, the lower was for the artificial neural networks, and the same was for the set 2. And also for R squared, the coefficient of determination of the validation set was the highest for the artificial neural networks compared to the other 2 methods in each set. And generally speaking, we obtained between 0.72 and 0.89 R-squared value. So this was the first way we validated the predictive performance. The second method was to find out if actually the models are real or not. Are the relationships obtained in these models real or not, or due to chance? So this was done by carrying out this idea of wire randomization. So instead of using models prepared with the actual responses, we randomized the responses among all of the formulations and then made predictions. And we notice here immediately that the predictions are meaningless. They are almost the mean of the actual predictions. They didn't change even. So these are the actual predictions on the y-axis. On the x-axis, the predicted values. We notice that most of them are very close to the mean of the actual predictions. So compared to the original models before doing this Y randomization, this proves that the relationships obtained in our original models were true, are not due to random chance. And finally, as a theoretical experiment, we wanted to test if the models, actually the predictive models, captured the significance of the formulation parameter, the PEG mole percent, because there is an optimal range of the PEG included in the formulations, above which or below which there will be a decrease in the performance. And it is always important to obtain this balance between the stability of the nanoparticles promoted by the presence of the PEG on their surfaces and their efficiency in vivo when they get internalized inside the cells, or how they are going to be internalized inside the cells. So what we have done here, because there is an optimal range above which the efficacy of the nanoparticles decreases significantly. What we have done here is that we have we have selected the formulations with a high percentage above the optimal range of PEG and decreased, like synthetically, changed the values of the concentration of the PEG to the minimal values in the optimal range. So if this fits with the observations in the wet lab that these optimal values should lead to better performance in vivo, we should see this in the predictions. And we see this actually in the predictions when we changed the PEG percent of the formulations to the optimal value. Actually, the predicted doses were shifted to a lower side, meaning they are more potent or more efficacious. This goes well with the general trend, the general observation of the optimal range of the PEG concentration. And the same also happened when we actually increased the PEG percentage of the formulations that originally have PEG concentration in the optimal range. We increased it to the maximum concentration, which is outside of the optimal range of PEG. And if this fits well with the wet lab observations, it should decrease the efficacy of the nanoparticles. And we can see this here with an increase of the predicted dose as compared to the actual doses. When we changed the value in the models, the features, we changed the value of the PEG descriptor to the maximum. So this has led to an increase in the predicted log dose, which fits again with the observations from the wet lab experimentations. So the conclusions of this work was that the dose of siRNA delivered by the cationic or ionizable lipid nanoparticles could be predicted successfully, provided careful machine learning modeling choices. Depending on the machine learning method and the validation set, R-squared of up to 0.89 could be achieved, which is reasonably good for in vivo predictions. Different machine learning methods resulted in different accuracies of predictions, with artificial neural networks resulting in the best performance in this work, and again, because they are data-driven. In this work, ANN outperformed the other methods of machine learning. And generally speaking, machine learning is a very useful tool when used properly. Machine learning is data-driven, so machine learning algorithms may have equal or different performance depending on the data set and the task that we are aiming to achieve. Machine learning saves time and resources, and very important at the end, machine learning can help us in our research. I thank my colleagues who participated in this work, and thanks to Professor Rania Hathout, we collaborated together from Ain Shams University. We collaborated together in many computational pharmaceutics projects, and indeed, thank you very much for your listening.
Thank you, Dr. Abdelkader. Thank you very much for your presentation. Is anyone having any question? Since not, then we will welcome our next speaker, Professor Klaus Michael Lehr. He just arrived.
Should be here.
Okay, I will introduce you. So it's my great pleasure to welcome my mentor, my PhD supervisor, Professor Klaus Michael Lehr. It's exactly 20 years since we first met, and since I met him, I would say my entire life changed, not only my profession or my career. Thank you. Professor Lehr is a professor at Saarland University in Germany, and co-founder and head of the Department of Drug Delivery at the Helmholtz Institute for Pharmaceutical Research, Saarland. Professor Lehr is co-author of more than 400 papers with more than 12,000 citations, an H-index of 70. The research interest of Professor Lehr is non-invasive drug delivery across biological barriers. Today, he's gonna discuss with us about the drug delivery on especially nano-mediated drug delivery for drugs, vaccines, and anti-infectives, and their role in the treatment of infectious diseases. So please join me to welcome Professor Lehr.
Good morning, ladies and gentlemen. Very pleased and honored to be here. Unfortunately, my voice is not in the best shape. At least the driver managed to get me here quite on time, I should say. Now maybe we are lucky and find my slides. Otherwise, I still have them on my computer. But I don't know where they're put. Somebody wrote me that they should be here.
Any specific name for the file?
Well, it has probably something to do with Kuwait, like many. Can we connect otherwise? Scientists are used to solve problems and are flexible. The name is Kuwait KIPC. Like any other slide. No, but I think we are in business. Right. Great. Fantastic, yeah. As I said, we try to be flexible. My voice not the best, but I'm here. Slides are running, so we can go. Thank you, Duno. It's really a great honor and pleasure. I'm so impressed by your city. It's first time, or country, state, that I'm here. Speechless. But that's not has nothing to do with the voice. I think we jump now into this and can discuss anything else later on. I'm very happy to be here and curious to learn more and also to contribute a little bit from that what I'm doing. Well, we deliver drugs across biological barriers. That's what I've been using to do since my academic career. I'm German, did my PhD in the Netherlands. Now I'm back to Germany and professor there. And typically when I go to international meetings, people ask me 3 questions. The first is, where actually the heck is this Saarland located? Well, probably you may recognize this is a map of Europe with in the middle Germany, and to the left in the south is, to the west is France. Somewhere you must cross the border between France and Germany if you want to, for instance, take a train, which you can do and go from Frankfurt, the big airport, to Paris. And where the border is crossed by the train, that's my hometown, Saarbrücken. And Saarland, which I have depicted here, is a small state. Germany is a federal republic. This is the smallest state of the 17 that we have. It's just 1 million of inhabitants. And it's a very nice view. You see, we are the state actually with most of the trees, most of the forest. This is an island in the tree called University Campus. It was a former military barrack, and after World War II, peacefully and creatively converted in a place of science and research. And the city of Saarbrücken is probably here where the floor is, and my house is about here, so it's just 5 minutes mountain bike walk, and then I'm there. And yeah, some of you have already been here, like Noah, for instance. So the second question that I often get asked, what is this Helmholtz thing that's often under my affiliation? Well, Helmholtz is, of course, a physicist that you probably know, and he gave the name, like Max Planck, like Leibniz, like Fraunhofer, other scientists and physicists, to societies that do research actively but are not university bound. So these are pure research institutions that do not teaching, but often from a certain level on, like me, head of a department, I'm co-founder of one of the institutes. You're also professor at the university nearby, which gives you access to graduate students and opportunity to teach. And you see Max Planck Society, I trust you must have heard about this, is the oldest and most prestigious ones. Fraunhofer Helmholtz is the second one here. And in all modesty, it is not the most well-known because it's relatively young. It was just founded when Germany got reunified again. But when you look at the right side at the numbers, we are by far the biggest association. And Helmholtz is supposed to do things in research that are supposed to be of national importance. We are sending ships to the North Pole. We are looking for structure of the matter, and we look for science. And in science, there is a new threat coming up. When our institute was founded in 2009, people were thinking, what is this guy doing? Why he is looking in infectious diseases? Everybody should care about cancer, Alzheimer, and these things, where the real threats and challenges are. Well, as we all know, in the past few years, that perception of infectious disease has somewhat changed. And ladies and gentlemen, that was just a viral infection. What I'm talking about, where maybe a bigger threat is, that's our bacterial infections. And you see what we have are the antibiotics. And some American politician had said in the 1960s, we can close the book of infectious diseases. We have solved all problems. We have the antibiotics. And we are used to take a box of 10 pills for $10 for 1 week, and then our infection is over. But unfortunately, that's less and less true. Because while there was no more new drugs, the bacteria have found that they can do something. They can get ready to defend this. This phenomenon is called antimicrobial resistance. And you see here the number of strains against antibiotics are no longer effective is increasing. While on the other side, the number of drugs, and even worse, the companies that are trying to make new drugs, they're getting less because it's not financially so attractive. And when we then look at some predictions of the World Health Organization, it may be that in 2050, if I make it, I will be about 90 years old, the chances are biggest to die from any of those diseases. So, high time to work on that. Not that we are saying we are going to solve all the problems, but we must do something, and we made a start. So, we joined our Department of Pharmacy with some professors with a bigger center of infection research and created this HIPS with a non-university research organization belonging to Helmholtz and located at Saarland University campus. That was my little introduction, that you know who that guy is and what he has been doing and where he's working on. Now I'm trying to tell you a little bit more what I'm actually doing. And the buzzword is biological barriers. Whenever we take a drug, whenever we use a drug, and I just got a last idea about siRNA from your talk, you cannot simply put this somewhere on the body or in the body. It has to reach a certain site, in this case, even inside a certain cell, where the cell is surrounded by barriers, but other barriers are the outer epithelial tissues. We can make technologies, and often they are based on nanotechnology. These are the carriers, but on the other side, we want to overcome the barriers to get the drug to the site of action. It's actually like a taxi driver. I needed to be at some place today, and I was in the car, and he had to bring me. He had to know where to drive and how to drive and all this. This is drug delivery across biological barriers, getting the drug to the site of action. It involves, of course, that you need to have a car, that you make a car, that you drive a car, but more important is actually design of the car. And if you want to successful in bringing something to its destination, you need to study these barriers in their biological requirements very thoroughly. That's what I and some other scientists of my generation thought we must do, and because the time was ripe that we could actually reconstitute tissues like intestinal epithelial cells. When I did my PhD, the CACO2 cells, which is now an industrial standard, just came up. It was possible to grow human skin, and we got involved actually in a time when we could actually do something on the lung. So for today, I will focus on just 3 barriers. This is a little bit a tapas menu, and I prepared 3 tapas for you. That's not going to be too much. And I will talk to you first about a very peculiar barrier. That's the bacterial cell envelope, which we need to overcome if we successfully want to overcome the problem of bacterial resistance. The others will be the skin, and then when we come to that, the last one will be the lung. And you will see I have a deep trust in the usefulness of advanced so-called complex in vitro models, often, whenever possible, based on human cells and tissues, because they allow us really what's going on when we intend to go across such a barrier. And this is a much wiser approach rather than making a famous nanoparticle and then giving it to a mouse, see what happens, go to church, even light a candle, and then hoping that you see the effect that you have been hoping for. But what if not? What can the animal tell you? It's too complex. So apart from all these ethical problems that we can reduce the number of animals for medical trials, we have, and that is my belief, that is an ambitious hypothesis that advanced in vitro models based on human tissues may be better predictors eventually for clinical trials and thus facilitate the movement on into clinical performance in the drug development process. So, talking about the bacterial cell envelope first. Why do we do this? Well, I told you we want to do something to combat the infectious diseases fundamentally. And what we used to do by giving antibiotics was actually aiming to kill the cells. So antibiotics kill the cells, the bacterial cells, and as you may imagine, bacteria don't like that. And in order to avoid that they become resistant, the more modest approach, the clever approach, the smarter approach is the so-called pathoblockers. In this case, you're not killing the bacteria, you're just trying to disarm them, if you wish, persuade them to not produce pathogenic factors and not be that dangerous. And because you're not putting them that much under pressure, you just destroy their weapons, the chance that they develop resistances are reduced. That is the concept, and there are some first drugs of that type. This is a kind of an antibody directed against a toxin that is produced by Clostridium difficile, has already reached the market, and I'm going to talk about more concepts in these lines and what we have to do in order to improve them. Well, one thing you have to realize, if drugs are supposed to act on bacteria and not to kill the bacteria, you probably must be able to reach the inner of the bacteria without destroying them. So it's not like you're killing a fly, patch, you smash it, or you take some surface active agents and you destroy the bacterial cell membrane. That's killing the bacteria, that's the same problem. You want to keep it alive, and in this case you must deliver a drug inside the bacterium. You must reach something which we try to coin as bacterial bioavailability. It's an additional pharmacokinetic compartment, so to speak, because what's outside the bacteria is not necessarily inside. And especially when you look at the gram-negative bacteria, these are CACO2 cells. This is ways how we can grow these cells and how we can predict what is going across these intestinal epithelial cells and become systemically available. When we look at the gram-negative bacterial cell wall, which is shown on top, then you realize this is not only an inner lipid bilayer like the mammalian cells as well. This is an inner lipid bilayer, then comes an hydrophilic intermediate layer here, and then comes another even asymmetric lipophilic layer on top of that. So this complex structure with active pumps and channels, that's actually where we want to go to the other side. And perhaps as a rationale to try to reconstitute that so that we have high capacity tools for screening and better understanding our molecules and delivery approaches. That was it was not the very first, but it was a system where we said, well, happy that we could make it. So we made a multi-layered system on such a permeable filter support. For those familiar with the Pampa concept, maybe that's similar. So we started actually with making different layers. And this is like if you were making a sandwich and you put first a layer of butter, then a layer of jelly, and on top a layer of maybe peanut butter. And if you were using a knife for that and you would smear it, you would destroy it all. So you have to make a layer by layer approach, and the last layer, as you can see, we actually deposited by aerosol technologies. We made this system, we characterized it, and then we were curious what it would be able to tell us. And then you see there's a correlation between what we call on the left side the permeability, and on the x-axis the in bacterio activity. That in bacterio means living bacteria. What really is, in this case, indeed, changing the bacterial activity, and what is the permeability. And you see, obviously, only drugs that can permeate and reach the inner side of action can do the job. And those who don't cannot do that. It's a very good indicative predictor. But that was a cumbersome system. So we thought maybe for a high throughput screening we can reduce it. And we actually had the trust, the idea that the main thing is actually the pores in this hydrophilic layer. So if the pores are important, maybe we can reconstitute that by making a hydrogel based on several polymers, and we tried many of them, and we found 20% starch. That's actually trivial. That's what you use to glue wallpapers on your wall. Was giving a very good predictivity. You see, we could generate now the data like in a CACO2 permeation model, and then we plotted them out against, again, the activity in vitro, and that was really happening in bacteria. And you see this for 27 drugs, a flawless, perfect predictability of the activity by the permeability through this membrane. So that looks good, and then we could also do together with some bioinformatics, some modern approaches, also based on machine learning, what I just heard, random forest analysis, and learning some new elements that could help to design the drugs in a way that they can do this better. We also did it a little bit more advanced, saying, okay, if we want to have the complexity of the cell wall, maybe we can also use these receptors. There is the concept of extracellular vesicles, so-called exosomes. You can make them from mammalian cells, but also from bacteria. Then make these membranes. It's a little bit more complex, and then you get also your predictivities. You see, I just want to go briefly onto that. It can be done. You get even a slightly improved predictivity, but for me, the best tool actually is this very simple high-throughput screening starch model. And we're also moving on further in computational tools. After in vitro probably comes in silico. So we are doing some molecular dynamics, very simple attempts to model, but molecule by molecule in an atomic resolution, the dynamic transport of the molecules going through that. That's taking a while until we can really move on with that. But this was my first appetizer. How was it? Good? You want to have more? I give you more. Let's talk about the skin. Skin is one of the oldest sites where people have applied drugs, and it's also a well understood, one think, biological barrier. So I'm going to talk about the skin, but not just about the skin. This is, let's say, the famous textbook conception with the mortar and brick model. Drugs go either the one or the other way, and that's certainly largely true, but for larger objects, larger molecules, or even particles, a different pathway comes in addition into the play. And these are appendages, especially the hair follicles. In fact, we were involved in some research that's now almost 20 years ago when it started. That was when nanotechnology was flourishing. People were discovering nanoparticles. It was said there's much more room on the bottom. Nanoparticles came up. Nanoparticles came up also as sunscreens, because now you could make titanium dioxide nanoparticles which are transparent and no longer white. People were putting that on the skin to protect themselves against ultraviolet light, but you don't want to look white, so you had an advantage with the nanotechnology. But people were also scared. Maybe if we put nanoparticles on our skin, they could perhaps go through the skin and end up in the brain, where we don't want to have them, and what happens. So there was big issues on nano safety, and it was a need to do that. And of course, we were happy to help out. We had some technologies, there were funding, and we collaborated with a group in Berlin, which is quite leading, Jürgen Lademann's group. He said, can you make us nanoparticles so that we can see what that happens when we put them onto human skin? And you see, there is an interesting relationship between the size of the particles and their follicular penetration. What you see here, and probably can recognize as a hair follicle, and here you see that nanoparticles, fluorescently labeled, penetrate quite deep into this hair follicle, in the cleft between the hair and the surrounding tissue. And this, for comparison, is just fluorescein, which is a much smaller molecule, but it cannot penetrate. Why so? So there was a theory developed that this has to do with two things. First, the hair surface is scalded, and on the other side, the hair is also moving. And you can even support this movement by applying a slight massage. And then you can even mathematically explain how this movement leads to the penetration of hair follicles in the skin. So, the good news from safety point of view, it's then inside your skin, but not really getting any further. And in fact, if you want to prove that on yourself, if you do some barbecue, which I guess you do it with charcoal, you go into the charcoal with your arms, and then later on to try to remove this by washing, you will see on the arms where your little hairs are, these little spots, dark spots may stay for a day or two. Okay, what can we do with this? I'm interested in the safety, but I want to deliver something, do something useful as a pharmaceutical scientist. So my colleague from the HIPS in Department of Vaccinology said, maybe we can reach the hair follicles associated dendritic cells. Every hair is a sampling probe for antigens that are around. This explains why we develop some antigens, how we can also desensitize, and maybe we could use that to achieve a non-invasive vaccination concept. And we did this, and as you can see by the data, that's an established mouse model that showed us there was a systemic, humoral as well as cellular response to some antigens that we had administered to the skin. You see then when we actually had improved the transparency, the migration, we really can do that. So very encouraging results. Unfortunately, the efficacy is relatively low, so with typical proteins, this is difficult. You would probably have to go on to more mRNA-based vaccinations. But let's move on first to another hair-associated disease. This is alopecia, but not in my case, where I can live with that. That's age-related and normal, and maybe it has to do and fits to my age, that's okay. But when you are a young person or a lady, you don't want to have this appear. It's not killing you, but it can be a nightmare. And this alopecia areata, that's sometimes in circles or sometimes affecting the whole body, is actually a complex autoimmune disease. The hair follicle develops an allergy against the hair, and that leads to the loss of hair that may be over after a couple of months, but maybe also taking longer. What you can do is you give immune suppressants. And because topically they are not active enough, you give them systemically. But then you suffer from side effects. So you're in a situation like this dog here that may be able to catch the bird, but what comes next, you don't want to see. So we thought maybe in this case we are dealing with a hair follicle that has no hair inside anymore, but maybe we can play a golf game and use these holes as a deposit for the nanoparticles. So that was where some of our colleagues from the PhD students got involved. They were ready to give away parts of their follicles. We also did some experiments on human bodies that had donated their body. It's very difficult to do this research actually on human skin, but we found ways to do that. And then you can see this is healthy skin on the forearm, and on the scalp in alopecia there's difference of the densities of hairs. And of course, the more hair you have, the more you can deposit per surface area, but per follicle it's actually about the same. It's not that much, but it's just a little bit, and you can use it. So we designed then some nanoparticles, and you will see about this particle more. It's a self-assembly based on a squalinated carrier. We could put tofacitinib, that's a so-called Janus kinase inhibitor, a very modern immune suppressant. It's used in the clinic, but the systemic side effects would be so severe. So we used that for that purpose, and we could see indeed that we got the association. We got more of the drug delivered actually into the follicular tissues. Oh, this is a little bit weak. So left side shows follicular tissues, and you see actually much more than interfollicular tissue, and you see that the nanoparticles actually did quite well. They're not as good as where you were dissolving the drug in acetone and DMSO, but as you can imagine, acetone DMSO is nothing that you wanted to put on the skin. So with the nanoparticles, you would have something that is indeed useful for a pharmaceutical development. But we were talking about infectious diseases. And infectious diseases actually also do occur in the context of the skin and the skin hair follicles. Sorry for these pictures, but in case of acne inversa or folliculitis decalvans, these are two most severe diseases of that type. Only surgeons can help. And here's the same thing. The question is, could we actually bring antibiotics into the hair follicles by using this phenomenon? And what we thought first, you remember my first slide, we need to design the carriers, but we need test systems to design the carriers according to the demands. We thought we need a system to study that, because on animals that's not the best way to do it, because also of the species differences and all that. Dermatologists and skin hair and hair researchers, they have developed methods where they work on floating hairs. Then you can study drugs that are stimulating hair growth and things like that. But you cannot study the phenomenon, what happens between the hair and the surrounding tissue. So we thought maybe we can, by using 3D printing, make a matrix in which we insert a hair. That is, for the first step, maybe easier than growing fresh hair follicles from induced stem cells and things like that, which currently is being worked on, but we wanted to progress a little bit further. So we did not even take cells, but we extracted hairs from active surgeons. Barbara Feldung is a very active colleague in the city, and she has many clients or patients that want to get their face lifted a little bit, and we get a little bit of this tissue and can extract the human hairs and plant them in. And then when you see the growth curves actually of the implanted hair in such a system, and the floating hair is about the same. So that looks promising. Now, if we go and take particles, can we actually see the accumulation of particles in this cleft? And we were honestly, to my opinion, more lucky than that we could hope that this really happened. But it's indeed the case. 200 nanometer particles will accumulate in this cleft. And when we challenged the system with bacteria, we also could observe that some staphylococci were really invading into this cleft and forming a biofilm in the area between hair and the surrounding tissue. That's great, because that's exactly what we wanted to study. And now if we want to treat such an infection by a drug like rifampicin, and we would be using floating conditions, you would of course see if you don't do any treatment, there's no effect. And if you give rifampicin in the same dose, it doesn't make much a difference if you have a free drug or a nano-encapsulated drug. But when you move on and you go to the model where you have the hair inserted in the follicle, then you suddenly see that in the hair follicle system, the killing effective is much higher, and the free drug is not able to penetrate, like exactly the same thing that we had seen in several cases before. So that's where we're standing at this moment, and that, I think, is an interesting point for further development. So you're still with me? I hope I'm still doing fine, voice-wise and time-wise. Then let's go to the last one. Lungs is personally my favorite. I fell in love with the lungs, so to speak, when I was a postdoc and had before worked in the Netherlands with Hans Junging and Douwe Breimer on intestinal drug delivery. Then I met a guy in Los Angeles, Vincent Lee, and on the same floor was a dear colleague, Kwang Jin Kim, and he was working in lungs, and that was by the time when John Patton was thinking about administering insulin via the lungs by inhalation. And this is actually a photo that John Patton, who I really admire for his pioneering work, showed to me. Look, and I realized that the lung is one of the biggest inner surfaces of our body, especially the deep lung. It is as large as a tennis court. It's about 100 to 140 square meter, when this is the deep lung. It's the same size like our gut as well, but the difference is one can compare the gut with a tube where the content is percolating through via several hours. In the lung, you take a deep breath and you're in. And then you hit what people call the air-blood barrier. Oh, before we come to that, this is Kennis Court applies to the deep lung where we have the alveoli, where the gas exchange is taking place. You see on purpose also a towel lying there. The towel, that is the bronchial tissue. These are our airways that we need to bring the air down to the deep lung, but this inner surface is actually much smaller. So when we are down there, then we're having really one of the most fascinating biological barriers that I know in our body. It's the what I like to call air-blood barrier. Not blood-brain, but air-blood barrier. And it is composed of an epithelium, a basal membrane, and an endothelium. And you see here in cross-cut a red blood cell, typically like 6 micron in this and about 2 micron in that dimension. And you will appreciate that the whole air-blood barrier with these 3 cell layers is extremely thin, less than 1 micron, and as large as a tennis court. And it must be extremely tight as well, because if it gets leaky, then you get an edema situation. And in fact, folks, that's what had happened in the COVID situation. When the virus is going deep down into the lung, you catch it with your bronchial tissue, but when it gets down and destroys that, that's where people then needed to this artificial breathing or and oxygenation, or eventually died because it was destroying that barrier. So we had talked about models and designed this to study what we can do with drug delivery. When you want to study transport across the air-blood barrier, you need a model, I would think, of this which is relevant. This is nasal or bronchial epithelial cells. When you go down these about 20 generations, the cells change in function and accordingly in shape. You get in a certain area cuboidal cells, and deep down in the lung, the cell must be extremely thin and flat and squamous. That's the alveolar epithelial cells. So the artist's eye could draw this like if you have a single alveolus, of which we have many millions in our body. You have type 1 cells, which are looking like a sunny-side-up fried egg, which cover the largest surface area, and you have smaller IT2 cells in between. And there was a cell line, and that's still often used. It's called A549. It's not bad at all. It's useful for many things. It's often used in cancer research because it's derived from lung cancer. But I would disencourage you to use it for transport studies, because you see the tight junctions between the cells are very poorly developed. And when you would be measuring with an electrode the resistancy, it is also very poor. We were then actually, by sending my first student when I had my own lab in Germany back to LA, learning the technique and coming to us, we were the first who succeeded with the primary culture of human alveolar epithelial cells. And they really look nice, as they should be. Their resistancy is extremely high. It's one of the tightest and at the same time thinnest tissues in our body, as I said. That is a primary culture. That's good because it's very close to reality. But if you want to use it frequently, standardize it, you want to have a cell line. So it took us more than 10 years until we came up with the first immortalized human alveolar epithelial cell line. It has the properties. It was called HELVI. It became available about 2017, right on time for the coronavirus research, and it's used, I would say, meanwhile, they're close to 100 groups probably. And then we also had some problems by the first users. They said it's not working reproducibly, and I thought we have to do something. So to shorten this a little bit, the details are all described in that CARE paper. when you're interested, we made a monoclonal selection. Actually, we picked a single clone with the best properties and developed this further. And that one we called ALO, and ALO is really giving fantastic, you see here, 4,000 ohm in resistancy. It also has the SARS-CoV-2 replication possibility. It has the ACE2 receptor and the TMPRSS receptor. So it's meanwhile also used by many virologists as a tool to study viral replication and things like that. You see that the barrier development of ALO is actually the best, even better than with the predecessor Helvi, and compares quite well with primary cells. There's a little bit of genetics. We can meanwhile say it's a type 2 cell with proliferative properties. It's not exactly a type 1 cell, but nevertheless probably useful in this context. So that's the cell, so to speak, the software. If you want to study pulmonary delivery, you also have to do something on your hardware. Because as opposed to studying blood-brain barrier or intestinal, the air epithelium is facing, the lung epithelium is facing air. We're talking about the so-called air-liquid interface, also referred to as ALI. And you want to deposit aerosols rather than pipetting drugs in a solution. And you need to develop systems, so this is one that we developed further where a company jumped in and developed something like a VitroCell powder chamber where you can deposit on one side. You can work with a regular inhaler and you can deposit just a very small fraction then directly on the cells to do research of that kind. It's also an interesting aspect of the lung that the lung is moving. Whenever we take breath, the diaphragma is causing a negative pressure, lungs is expanding, and that causes a lateral stress on this epithelium. And you see this is an in vitro system. You see it moving. It moves about 10 times in a minute by applying cyclic pressure, so you can do things to mimic the effects even more. I go not too much in details. It's interesting, especially in inflammatory diseases, not for all purposes, but it's good to have these options. I want to come back to the infections. And what we actually want to have is something like an in vitro patient, like this, not too old yet guy, who might be suffering from mucoviscidosis, cystic fibrosis, and inheritary disease, leading to production of poor quality mucus, then leading to a chronic biofilm infection, typically by Pseudomonas. And the problem is you need to apply antibiotics on a daily basis just to control that, but you cannot really get rid of the infection. You cannot eradicate it. So, and we thought if we want to model that, we have to realize three aspects. We need a lung model, human-based. We need to put and grow biofilms on top of that, and we need to have the possibility to reproducibly deposit aerosols. So binary combinations of these elements were all described, but having all the three things together, that was not yet there. And in all honesty, it were about 3 PhD theses until we finally came to a first protocol, which I'm sharing now with you. That was that we have to grow the alveolar cells separately in the same lab, but in the in an incubator next door. Some people are reluctant to do that, and that's absolutely understandable. But meanwhile, we have this under control. We grow the bacteria on the other side, and when they start from being planktonic and form a biofilm, also after about 7 days, then we marry the two systems, bring the biofilms on top by pipetting so-called microclusters of the epithelial cells. And that leads to the situation that they're not immediately killing, and you can do useful experiments actually over periods of several days. We have so far managed up to 3 days. The dream would be to 3 weeks, maybe even longer, that you really have a chronic infection model that you can see how these things are going. And the good thing is you can generate readouts that are telling you something about the bacteria, and you convince yourself that under repeated application of tobramycin, actually the amount of bacteria is going down. It's not completely eradicated, but it's getting less. You can see that you can maintain the viability of the host cell. You can see that you can even restore the barrier properties. This is this TER, which under tobramycin comes back, so the young lung is really getting better. And you can also study some of these virulence factors that we want to control and to suppress by means of these new drugs. The next step, and that's exactly where we're currently at, the way how we transfer the biofilms is still a little bit rudimentary, and we thought maybe we could do this better by, again, applying bioprinting. And again, that was also not so trivial. If you want to develop inks where you can grow bacterial biofilms and write and print something, you must do the right rheology that you can really print something and write the words of the letters of our institute, and then when you leave them, the bacteria grow in that shape. You can put this on cells, and the cells will survive that, and you can then switch from E. coli also to Pseudomonas aeruginosa. And the good thing is that you see when you compare native biofilms and native planktonic, you can convince yourself you have a different susceptibility to antibiotics. The native biofilms are more resistant. In this case, the word is tolerant towards the antibiotic. And when you do the same thing with 3D printing, you perceive the same properties. So it's important actually to have that. Good. That's all about the models. And now I told you, and I guess otherwise you wouldn't believe me in reality, I'm not a microbiologist or cell biologist. I'm a pharmaceutical scientist. I want to deliver drugs. So we thought if we need to have the application of one of these biofilm suppressant agents that are inverting with the quorum sensing, Noah, you know these type of drugs as well, and at the same time we want to also administer drugs like tobramycin that eventually really can kill the bacteria, we need to actually have a carrier that is capable to incorporate the two drugs with different properties in the same system and also capable of being nebulized. And we succeeded in making these nanoparticles. I'm not going into details. We could see, and that is important, and we still don't know how it works, when they are landing on the surface of such a bacterial biofilm, the red dots, which are the carriers, they really can penetrate through it after landings, and that is the key. They can really reach the bacteria and then change something. And what the result is actually, when you have tobramycin only and you try to eradicate an infection, this is the so-called minimally biofilm eradication concentration, and even at the highest concentration, there are still some survivors. That's exactly the clinical situation. You can control but not eradicate the infection. When you combine it with one of these quorum sensing inhibitors, hooray, at relatively high concentrations, you suddenly can eradicate it. But when you then put all the two in the nanocarriers, you have not only a doubling, you have about 30 times improvement. So you can eradicate the bacteria by using 30 times less of the antibiotics. And although this is but I think relevant in vitro system, yet this is an encouraging result. So we got some nice papers, some awards for it, and that's about where we have been. If I still have 5 minutes, yes, then I continue giving you an outlook that maybe connects a little bit to what you have been presenting, the nucleotide delivery thing. We are currently involved in a project that actually is about to transfer the concept of CRISPR-Cas. I guess you know that, Nobel Prize awarding gene editing system that was kind of invented by bacteria to defend themselves against phages, to transfer that by using other CRISPRs to mammalian cells so that we had a way to fight against human viruses. And what you need to do then is a dual delivery of 2 nucleic acids. You have to deliver a messenger RNA as well of the CRISPR, as well as a guide RNA that the CRISPR knows what to do for. So dual delivery, polynucleotide delivery is the thing. And exemplarically, we can show here that with some nanocarriers, we can encode both red and green fluorescent dyes, and in the same carrier, we have an efficient co-transfection in the same system for the both systems. We're also working on some new polymers to do this. These are peptide-based biodynamers. They can dynamically assemble and disassemble. The most important property is that they're extremely non-toxic. You see they're extremely well tolerated by cells compared to other polycationic things, which I think is probably a most crucial thing. We can use lipids, which are typically used for these messenger R systems, but the lipid systems are, and I guess you have been talking about this, are extremely sensitive. We must keep them at low temperatures, and you probably are not so well advised if you try to nebulize these systems to make an inhalable aerosol. So we are actually working therefore on lipid-coated polymeric carriers, which provide us kind of the best of the two worlds, stability by the polymer and the needed and useful properties by a lipid coating around. And you see It's shown here that we can successfully nebulize these systems and by that achieve hopefully dual transfection with 2 different nucleotides on target cells on the loan. So, ladies and gentlemen, that's what I had prepared. The chairperson is already getting nervous, but I hope I was not overdoing it. This is our institute. We were last year hosting the International Society of Aerosols in Medicine. It was fun to do so. If you ever have a chance and want to visit Germany, come to us. We have every year a HIP symposium where we focus on various aspects of infection research. Thank you very much for your interest and for the opportunity of being here.
Thank you very much, Professor Lair. And we will move, for the sake of the time, for the next speaker. And maybe if there is discussion or question, we can discuss it during the coffee break. Thank you very much. Last lecture for this session will be held by Dr. Dunya Bastaki, Head of the Drug Registration Department in the Ministry of Health. Welcome, Dr. Dunya. The lecture will be related to pharmacogenetics and drug registration.
The other one is not good? Oh. Mm-hmm. Okay. Yes. Okay. Good morning, everyone. It's my great pleasure to be here with you in today's conference, and it's really also my pleasure to see my dear mentors and colleagues today in the room. I hope my presentation will not be a long one, and it will mainly highlight on integrating pharmacogenomics into evolving drug registration guidelines. So the agenda will cover the pharmacogenomic definition, the molecular basis of drug response, consequences of genetic variations, pharmacogenomic information of the drug label, regulatory aspects of pharmacogenomics in drug discovery and drug approval process. So the pharmacogenomics is defined by the ICH. Of course, all of you are aware of the ICH, the International Conference on Harmonization, as the study of variations of DNA and RNA characteristics as related to drug response. Simply, it's using patients' genomic information to improve efficacy and safety of prescription drugs. So the pharmacogenomics can play an important role in identifying responders and non-responders for medications, avoiding adverse events, and optimizing drug dose. So to take a quick look on the molecular basis of the drug response, the genetic variation can affect the drug pharmacokinetics as well as the pharmacodynamics. The genetic variants that would affect drug metabolizing enzymes or drug transporters would definitely affect the pharmacokinetics in terms of absorption, distribution, metabolism, and excretion. Also, the genetic variants that affects the receptors, ion channels, signaling cascades, and unintended targets would affect the pharmacodynamics of the drug, affecting the pharmacological effect, efficacy, and toxicity. As you know, there are more than 50 cytochrome P450 enzymes in human body. 5 of them involved in the metabolism of around 90% of drugs. The ones with the most inter-individual variability and activity are the CYP2C19, CYP2C9, and CYP2D6. And if we're gonna translate it into phenotypes, we have as well the ultra-rapid metabolizers, extensive metabolizers, which are the normal metabolizers, intermediate metabolizers, and poor metabolizers. As per the three enzymes of question that we were talking about, CYP2C9, CYP2D6, and CYP2C19, as you can see, the majority of the population lies between the extensive and intermediate metabolizers, whereas poor and ultra-rapid consist of poor, mainly found in Asians and African ancestors, having around 2 to 20% of the population for CYP2C19, and the ultra-rapids around 30% for CYP2C19, where those subpopulations need to be considered in our therapy decision and in our dosing and targeted therapy decisions. Of course, the CYP2C9, mainly the drugs that are affected are warfarins, phenytoins, and NSAIDs. For CYP2D6, opioids, antipsychotics, and beta blockers. And for CYP2C19, tricyclic antidepressants, clopidogrel, proton pump inhibitors. Those are some examples of those. So many different alleles or variants exist in the population. If we can take a look on the CYP2C19 alleles and what are their impact, the most common allele is the STAR1 allele, which is considered as a normal allele. STAR17 is considered a gain of enzyme function allele. STAR2, 3, 4, 5, and others are considered a reduced or loss of enzyme function allele. The database for FARM-VAR is really a very good database that we can get what are the different types of alleles for each variant. For the genotype activity correlation between the phenotype and the underlying genetics, for the extensive metabolizers, you can see at least one normal functioning gene will be available. Of course, this is the majority of the population. For the ultra-rapid metabolizers, we can see extra copy or copies of gene or increased gene expression. For the intermediate metabolizers, there would be one, the white box, one non-functioning and one reduced functioning allele. And for the poor metabolizers, there are two non-functioning alleles. So a quick overview on the warfarin mechanism in normal metabolizers and poor metabolizers. For the elimination of the warfarin, as you know, warfarin would target the VCORC1 to reduce the clotting in patients. They use the CYP2C9 in order to eliminate from the body and convert into an inert metabolite. In poor metabolizers, where the CYP2C9 are not functioning properly, there would be a high dosing of warfarin in the blood would result in toxicity and uncontrolled bleeding. On the other side, for prodrugs such as codeine, the normal metabolizer would change the codeine as a prodrug with the CYP2D6 into the active metabolite as morphine and exert its effect on the opioid receptors and result in pain relief. For the poor metabolizers, where CYP2D6 is not working efficiently, there will be, of course, a lack of efficacy and little or no pain reliever. So if we would compare the effects or the consequences of the genetic variation in an active drug versus a prodrug, such as warfarin or codeine, for the poor metabolizers, you can see that it has a reduced elimination and increased toxicity, whereas for prodrugs, poor metabolizers will have decreased activation and reduced efficacy. For ultra-rapid metabolizers, they will show increased elimination, reduced efficacy, and for the prodrugs, it would show increased activation and increased toxicity. So the normal practice of one dose fits all is not anymore the best practice. Now we are, as you know, we are moving towards the precision medicine, the personalized medicine, and actually now more than 30% of the FDA-approved drugs are personalized medicines. If we go for the standard framework for the one dose, one diagnosis, you can find standard responders, which are the majority of the patients. They would respond to the drug. But also you can find non-responders, and you can find those with adverse events. In case of precision medicine, where we use the pharmacogenomic data and evidence, and we can then target the dose to standard dose or higher dose for those that have altered metabolism, or we can recommend an alternative drug. So let's go to the US FDA new regulations on the pharmacogenomic info in the drug label. Of course, the guidelines have been updated more than one time, but the most updated one cover more than 500 drugs that have pharmacogenomic information in the drug label. The drug labeling may contain information on genomic biomarkers and can describe the drug exposure and clinical response variability, the risk for adverse events, the genotype-specific dosing, the mechanism of the drug action, and polymorphic drug target and disposition genes, and the trial design features. So, as you know, in the US as well as Europe, the therapeutic area that mainly you can see pharmacogenomic data in their labels are the area of oncology. And as you know, most of the targeted therapies are under the oncology therapeutic area. So where can we find the pharmacogenomic data in the leaflet? As you know, the leaflet has sections of indication, adverse events, contraindications, clinical studies, using specific population, box warnings, and others. Actually, it's found everywhere. So you can find it in any section, depending on the source of data. So actually, you cannot look into some sections to detect, but the most sections that we can see them in is the clinical pharmacology, clinical studies, indication, and usage. There are two important tables on the FDA website covering the pharmacogenomic information and data. The table for pharmacogenomics biomarkers and drug labels. The table lists therapeutic products and pharmacogenomic information found in the drug labeling. The labeling for some, but not all, of the product includes specific actions to be taken based on the biomarker information. This is how the table looks like. You can search for whatever drug you want, and it will get you information on the therapeutic area, the biomarker that is related to the drug, and the labeling sections where you can find pharmacogenomic data or recommendations for patients and for healthcare professionals. When you click on those, okay, it's not working. Okay. When you click on those, you can see it in details. Let me show you on the second slide. Yeah, it will open up the page where you can find the drug labeling, genomic information, and the recommendations needed. Okay. Another table is the table of pharmacogenetic association. And the table lists the pharmacogenetic associations that are related to drug metabolizing enzyme gene variants, drug transporters gene variants, and gene variants that have been related to a predisposition for certain adverse events. It's divided into three sections. The first section is the pharmacogenetic association, for which the data support therapeutic management recommendations. The other section is for pharmacogenetic associations for which the drug indicates a potential impact on safety or response. And the third section covered the pharmacogenetic associations for which the data demonstrate a potential impact on pharmacokinetic properties only. Also, this is how the table looks like. For example, this is section 1. Also, you can find the drug, the gene, the affected subgroups, and the description of the gene-drug interaction. So a study published in 2014 analyzed the data found in the drug labels for the FDA drugs approved, and they found that among drug labels with pharmacogenomic information, the clinical validity evidence stands for 36%, where the clinical utility evidence is 15%, and the clinical recommendations account for 51%. So, as you know, there are some challenges for the FDA labeling. One of them is the lack of solid evidence. As you know, it's a new area, and we're getting to know day by day more data is coming up. I will also cover some of the regulatory aspects and how the registration process now demands the pharmacogenomic data to be submitted in the registration or the drug approval dossier. But up to now, there are some drugs that lack solid evidence. The limited access to testing. The pharmacogenomic testing or genomic labs are not usually accessed by all due to the high cost and due to also the unavailability in most of the healthcare institutes. And we have to know that the FDA balances need to communicate important information without being too prescriptive. So a mention in the FDA label does not imply clinical validity or utility of a pharmacogenetic test. Sorry. So along with the FDA label, we cannot consider it as the main sole reference or resource for our pharmacogenomic information. So as a healthcare professional, we have also to look for guidance or references that would cover the clinical validity as well as the clinical utility. The CIPC is one of the authorized softwares or database, which is the Clinical Pharmacogenomic Implementation Consortium. It is really a big source of information that is highly reputable and it is authorized as well. It evaluates evidence supporting prescribing changes, the clinical utility. They have up to now 26 guidelines published covering around 154 drugs. It is peer-reviewed, evidence-based, updated, and uses standard format and detailed gene-drug clinical practice guideline. The CIPC guideline focus on how available genetic test results should be used in practice to optimize the drug therapy rather than whether tests should be ordered. So the FDA label would clearly mention this test should be ordered for patients with this criteria. The CIPC will give you more clinical information on how to utilize the data of the genomic test and how to use it in the clinical practice. Also, one of the most important references and resources is the PharmGKB. The PharmGKB is a pharmacogenomic knowledge base that evaluates evidence of gene-drug association or the clinical validity. It interprets the FDA label, the sections, and I will show you also there are other useful information that you can use. It has parts that cover clinical guideline annotations, drug label annotations, FDA drug label annotations, and curated pathways. So what's the difference between the drug label annotations and the FDA? Here it covers also other health authorities such as the EMA for Europe, such as the Canadian authority, the Japanese authority, the Swiss Medic is added recently. So you can even compare between the different drug labels and the different recommendations between the different health authorities. Here also this section that covers the curated pathways, here it provides diagrams on mechanism actions and on the gene of question pathways, and it's even very useful. It is open, free for access, and anybody can go through it. So the drug labels in the PharmGKB, the interpretation would come up with whether this testing is required or recommended, actionable or informative. And as you said, it covers 5 agencies: the FDA from the US, EMA representing Europe, Swiss Medic, Switzerland, PMDA, Japan, and HCSC, Canada. And if we look for, this is the drug label overview. As you can see, it shows the sections related to the FDA, EMA, Swiss Medic, HCSC, and PMDA. And as you can see, it's not really, sometimes they don't agree on the same recommendations. So sometimes the FDA required testing, the EMA also required testing, but PMDA does not require testing. So still we have this kind of lack of consistency between the different authorities, and it's always dependent on the clinical judgment of the healthcare professional as well as the other guidelines published. So the dosing guideline in the PharmGKB is extracted from guidelines such as CIPC, the Clinical Pharmacogenomic Implementation Consortium, the DPWG, which is also a very good reference, Royal Dutch Association for the Advancement of Pharmacy, the Pharmacogenetic Working Group, CPNDS, the Canadian Pharmacogenomics Network for Drug Safety. So it have guidelines from those entities, as well as the drug labels from the health authorities. The PharmGKB levels of evidence, it starts with level 4, 3, 2B, 2A, 1B, and 1A, and if we go up, it is the highest level of evidence. So if a level comes up with a level 1A, it means that there's a CIPC guideline or with known clinical implementation. 1B, there's a strong evidence but need replicates. 2A, the variant is in PharmGKB VIP database. 2B, variant not in PharmGKB. 3, single significant, not yet replicated, or non-significant in multiple studies. And 4, case report, non-significant study, or in vitro data. So it's also important to check for the level of evidence. We have a tool which is made in Kuwait too, and I'm very, very proud of DASMA and colleagues for releasing and creating such a tool for pharmacogenomic testing. The tool created by the SMAN Institute is called PharmaQ. It's an open access, and it's really, really easy to work with. You just need to have the genome variant file. So the tool enables physicians and geneticists to carry out preventative pharmacogenomic testing, which help to customize medication prescription by having a patient genetic information in hand. The tool requires that the patient has his whole genome sequence, a service offered at the SMAN. The physician will input the patient's genome variant file, the VCF file, into the PharmaQ and generate a personalized pharmacogenomic report. So if there is a drug of question, or if the patient has a history of non-responding or an adverse event, or it's a targeted therapy, the healthcare provider can request a genomic test, and based on the genomic test, he will have the VCF or the genome variant file that would simply be uploaded on this front page here. Okay, and upon uploading the VCF file, it will come up with the analysis report. It's a personalized per patient, a personal pharmacogenomic report that covers the genes, different variants, the genotype, the allele functionality, the phenotype, and the clinical recommendation. So here I would use a usual dose. For example, in this case, I would adjust the dose. In others, I would also need to adjust the dose. It will give you clear recommendation of each drug and how to work with it. Here's the interpretation, second page. For example, let's say the CYP2C19, it gives a recommendation on this patient and on certain drugs of question, if there's any recommendation needed for like dose adjustment, or even to avoid such drug in such patient. Okay, so now I would turn to Now I would move into the area of regulatory, which is my zone of comfort and my area of expertise. As you know, I'm a regulatory authority specialist, and this kind of pharmacogenomic data and this kind of information now is highly needed when we regulate, or when we look into the new drug applications or new drug files. So the pharmacogenomic data submission actually started in 2006 with a joint guideline between the FDA and the EMA. The guide was named Processing Joint Food and Drug Administration European Medicine Agency Voluntary Genomic Data Submission. It was voluntary at that time. within the framework of the confidentiality agreement between the two agencies. The FDA and the EMA, I wrote EMEA, which is the old name because it was in 2006 still EMEA, issued guidance for industry named Pharmacogenomic Data Submission and Guideline of Pharmacogenetic Briefing Meeting. Respectively, both documents encouraged the voluntary submission of genomic data by the sponsors to the agencies. Also, the FDA now in March 2023 issued a draft guideline on the pharmacogenomic data submission in the registration file. So it covers the INDs, which is the investigational new drug application. It covers the new drug applications, the NDAs, and also the biological license applications based on the FDA criteria and regulations. So now it should be included in the submission to say, and here how it should be submitted. So let's say for INDs, investigation of new drugs, some areas need, some parts need to be submitted as detailed reports. Other, we can rely only on synopsis, no need for a full detailed report. Of course, it's still a draft guidance, not yet finally published. The synopsis or the detailed report in association, data submitted to the NDAs or BLAs should be referenced in relative sections of the submission. As you know, the submission dossier, which is the CTD file, has different sections, and each section is related to whatever kind of information, study, or document to be included in the specific location. So where to put this kind of pharmacogenomic data in the CTD structure? It's in Module 5 in 5.3.5.3, and if the applicant would have multiple data, so the FDA encouraged summarizing these data in relevant submission summaries as the integrated summary of safety or the integrated summary of effectiveness as appropriate. So also the pharmacogenomics is really important in the area of pharmacovigilance and monitoring of safety of medication post-marketing. The EMA has a really brilliant guideline called the key aspects for the use of pharmacogenomic methodologies in the pharmacovigilance evaluation of medicinal products. Here's the link for the guideline. It shows how to evaluate the pharmacovigilance related issues for medicinal products. For medicinal products with pharmacogenomic associations. I think for pharmacovigilance specialists and people working for pharmacovigilance, you all know that. Okay, it worked? So, okay. Okay. All right. So anybody working in pharmacovigilance would know that the causality assessment is being a broader science now. And if we want to perform a proper investigation or causality assessment to show the association between the drug and the adverse event, one of the important aspects we have to look into is the genetic variation between patients and to dig more into the pharmacogenomic data. It also shows how to translate the results of these evaluations to appropriate treatment recommendations in the labeling, as we said, the types of genomic biomarkers relevant to pharmacovigilance, and the most importantly is how to implement pharmacovigilance activities and risk minimization measures. The RMMs or the RMPs now have a big section on how to minimize the risk in association with the pharmacogenomic data or the genetic variation. So this is something that we look at nowadays. It was not basically used before, but it's now one of the core methods that we use for causality as well as for risk minimization measures. So to wrap it up, when to consider pharmacogenetic testing? We should consider it always for certain drugs, such as targeted therapies, personalized medicines, and for those who develop for those drugs that develop to work in patients with specific variant or variants, or drugs which is not effective in patients without variant. Also, testing requirements clearly, if the testing requirements is clearly stated in the label, or to avoid serious life-threatening adverse drug reactions, or if there's a personal history of ADR or lack of response for the patient, and also I would add on upon clinical judgment. And a summary of my presentation. Okay, so genetic variation can impact pharmacokinetics and pharmacodynamics of drugs. The purpose of pharmacogenomics is to use genetics to optimize drug therapies, maximize drug efficacy, and minimize adverse drug reactions. The pharmacogenomics is the fundamental element in precision medicine. And the pharmacogenomic information in drug label, along with authorized references as CPIC and PharmJKB, need to be considered in therapy decisions. And we need to highlight always on the importance of the regulatory initiative to promote the use of pharmacogenomics. I ended up, and thank you very much for your attention, and it's really my pleasure being with you today. Thank you.
Thank you very much, Dr. Dunya, for your presentation. I think you need now a coffee break. So let's go for 10 minutes coffee break, and then we will resume our session, inshallah. Thank you very much.
very minimal number of people. Hopefully we'll get more and more. I know you guys are expecting Dr. Dalal Tawil to present, but we agreed I'll moderate for the next two presenters. So welcome back. This is section 2. We'll give it more of a clinical pharmacy tweak. So our first presenter will be Dr. Amal Al-Harbi, and she will be taking us through her presentation on pharmacovigilance communication in Kuwait, our current situation, and where our future direction will be. Little brief about Dr. Amal. Dr. Amal is currently a member of Kuwait Pharmacovigilance Office and also the National Antimicrobial Consumption in Kuwait. As part of her work experiences, she has been working in Drug Inspection Administration. She also worked as a clinical pharmacist in Farwania Hospital. Dr. Al-Harbi has achieved her PhD in the University of Hertfordshire in 2023. Previously, she did a master's in clinical pharmacy. That was in the same university, 2017. And she has received her B.Pharm in 2013 in Kuwait University. Dr. Al-Harbi has, in her experiences with research, she has performed systemic literature reviews, mixed method research, interpretive phenomenology, I hope I said that right. And her research also includes, of course, the pharmacovigilance, antimicrobial stewardship implementation, medication adherence in asthma, as well as medication management in dementia. Please help me welcome Dr. Amal Harbi for her presentation.
Good morning, everyone, and thank you all for attending today. Today I'll be presenting about the pharmacovigilance communications in the state of Kuwait. As this was part of my PhD journey, I would like to acknowledge the efforts of my supervisors from Kuwait University, Dr. Fatma Saleh, and from the University of Hertfordshire. So pharmacovigilance is the science and activities relating to the detection, assessment, understanding, and prevention of adverse drug reactions or any other drug-related problems. The need for pharmacovigilance arises from the fact that in the pre-marketing phase, clinical trials usually use a selective of patients that underrepresent the actual population. Unfortunately, the recognition of pharmacovigilance had occurred after a tragedy where in the 1960s, 10,000s of infants were born with congenital deformities due to maternal exposure of thalidomide, a drug once promoted as being safe in pregnancy. The aftermath of the thalidomide disaster has resulted in multiple regulatory changes, one of which was the establishment of the International Drug Monitoring Center in Uppsala, Sweden. Kuwait has become a full member in the center since 2021. So pharmacovigilance communications refer to the active dissemination of updated information related to the benefit-to-risk balance of medications. These information could be disseminated in the forms of letters to healthcare professionals or the patient. They could be posted on websites or sent through emails or mails. But why do we need communication in pharmacovigilance? Simply because without an effective communication, pharmacovigilance would not achieve its goals in terms of protecting the patient and safeguarding the public health. Failure to communicate might lead to the spread of misinformation, mistrust, and misguided actions. Now, pharmacovigilance communications do not only focus on transparency and ethical fulfillment, but usually they have expectations in terms of their outcomes, for example, facilitating changes in beliefs, behaviors, or attitudes. So there has been a number of studies and systematic literature reviews who evaluated the impact of these communications in the clinical practices, and they found that there's not always adherence to these recommendations. Above that, they found an evidence of unintended effects, meaning that, for example, when the FDA had issued its recommendation regarding the use of selective serotonin reuptake inhibitors in the pediatric population, it recommended to monitor the patients for the first month of treatment initiation. However, they found that physicians were stopping the use of SSRIs in the pediatric population as a result of the communication. There was also evidence of spillover effects. Using the same SSRIs examples, they found that some healthcare professionals were stopping the use of this medication in the adult population, although the alerts and the recommendation never targeted these patients. So we have conducted a systematic literature review that was published as 2 articles in Research in Social and Administrative Pharmacy, and we have found that the implementation of pharmacovigilance communications in the clinical practice depends on a multiple level process, starting from the creators of the alerts or the message, whether it was the regulatory agency or the pharmaceutical company, their dissemination strategy, the message itself, its clarity, its content, and then the receivers of these messages, whether the healthcare institutions, for example, their policies and interpretations, the healthcare professionals, and the patients. As there was no previous studies that have evaluated pharmacovigilance communications in Kuwait, we sought to evaluate these communications within the Ministry of Health. To achieve this, we have conducted a multi-phase research approach. Starting with phase 1, we targeted Kuwait Drug and Food Control, or KDFC, specifically the Pharmacovigilance Unit, or the PV Unit. This is because up until October 2023, the PV Unit was responsible for pharmacovigilance activities within the state of Kuwait. This was a convergent mixed method approach where we analyzed documents and conducted face-to-face interviews. From this phase, we used one example, which was the valproate teratogenicity communication that was sent to the MOH hospitals. So we used it in phase 2, in which we targeted healthcare professionals through focus group discussions and survey distributions. And then in the third phase, we aimed to explore the patient experiences who were using valproate. Now I will give an overview of the main results. So as I said previously, the PV unit is responsible for the creation of pharmacovigilance communications in Kuwait. To achieve this, they usually receive information whether from their ADR reporting system, online system, or through other methods like international regulatory agencies. They usually assess the information and then take action in response to this information. In addition, they regulate the pharmacovigilance activities of the pharmaceutical companies, whether the early phase activities or the post-marketing activities. We have found that most of the sources that the PV unit used to create these communications were related to international regulatory agencies, specifically US FDA, the UK MHRA, the WHO, and the European Medicine Agency, followed by the local marketing authorization holder within the country. The marketing authorization holders are obligated to submit any updates and safety information to the PV unit within 15 days if it was a serious case, or within 90 days in non-serious cases. The PV unit had also conducted multiple action in response to this information, for example, distributing risk minimization measures with the withdrawals of certain medications from the market. But the most repeated action was informing healthcare professionals about the updated information. The tools that they used to send this information included Dear Healthcare Professional Letters, or DHCP letters, and Kuwait FDA newsletter. This is an example of one issue of Kuwait FDA newsletter, which is disseminated every 2 months by the PV unit to the Kuwaiti hospitals. The decision that they take in terms of deciding whether they use a single tool and which tool to use, or use a combination of tools, depends on the criticality of the case and whether or not it needs to be immediately communicated. For example, if it's a critical case that needs to be immediately communicated, the PV unit would send a DHCP letter itself. If it was a less critical case, the pharmaceutical company would be expected to send these letters. Most of the repeated letters was related to the SGLT2 inhibitors. We found some inconsistencies in terms of the content of the letters. For example, only 15% of the letters included quantitative information about adverse drug reactions. Surprisingly, none of the letters included any quantitative information about the medication efficacies. Almost all of the letters were distributed in English. Only few of the letters that were aimed to the public were distributed either in Arabic or Arabic and English. This is an example of DHCP letter that was previously distributed to the Kuwaiti hospital. It was about the valproate teratogenicity. It included risk minimization measures such as the prescribing checklist and the patient guide. It was also required from the healthcare professional to distribute this patient card to the patients and to obtain a consent from the patient, acknowledging their understandings of the risks associated with valproate. Now, although most of the letters were aimed to healthcare professionals, actually none of the letters were sent directly to the healthcare professionals at the patient-facing level. They were actually manually distributed to the directors of the health areas, then to the directors of the hospitals, and expected to be distributed to the head of the departments. Currently, there is no process in place to monitor the implementation of the recommendations in such communications. Now, for the healthcare professionals, generally we found a positive attitude from the healthcare professionals regarding updating their knowledge about drug information. However, there were lack of awareness regarding the existence of the PV unit and the tools that they use to disseminate the information. And this was surprising, as I previously said, one of the tools was actually distributed every 2 months for years to the Kuwaiti hospitals. The barriers that they mentioned regarding why do they not receive this information was related to the manual distribution and related to the large amount of random information that could mask pharmacovigilance-related information. And of course, there were some groups of healthcare professionals, like the nurses, that felt that they were excluded from any drug safety-related communications. Regarding the valproate-related safety communications, only 2.6% of the participants in their survey knew about these recommendations, even though they knew about the teratogenicity of valproate. So this was reflected on the impact of this communication in the clinical practice. So we saw law adherence for the recommendations of KDFC. For example, we only saw less than 40% of the healthcare professionals providing female patients with written information, and less than 40% counseled female patients about on contraceptives use. There were multiple barriers to implementing the valproate recommendations in the clinical practices, one of which is the lack of their awareness about existence. But we also now noticed throughout the studies that we conducted that there was a sense of responsibility transfer between the healthcare professional groups in terms which healthcare professional group should implement these recommendations. Now, for the patients, we had interviewed a total of 6 patients from the ages of 20 to 48 years old to understand their experiences. 4 of these patients were married. 3 of them had previous experiences with pregnancy, and 1 was pregnant during the interview. All except 1 patient were not aware about the teratogenicity of valproate, although they have been using it for a long time. All of the patients indicated that they did not receive written information about its risks. Neither they were asked to sign acknowledgements to reflect their understanding. We have also noticed lack of knowledge in general related to the valproate among the patients. Again, as I said before, they have been using it for around 10 to 20 years. The patient explained that this was related to the fact that their parents were responsible about the drug-related information because they were diagnosed at a young age. So this type of information would be collected by their parents. They did not have the opportunity to take the information from the healthcare professionals later in life. And some of the patients did not have the interest to know about this information. For example, fearing from knowing negative information. We also found that the patients had wrong perception whether overestimating or underestimating the risks of the medication, and this was attributed to the miscommunications between the patients and the healthcare professionals. Regarding the recommendations, currently the PV office or the pharmacovigilance office has been established. We are trying to create communications with the healthcare institutions and to create focal points in these institutions. The primary goal of the focal points is to promote the adverse drug reaction reporting, but they could also have a secondary practice, which is monitoring the implementations of the pharmacovigilance recommendations. There should be promotion of awareness to the healthcare professionals to increase their awareness regarding the pharmacovigilance activities and the pharmacovigilance communications. There also should be a clear information given to patients who are using valproate, preferably on an annual basis, and preferably to have a pre-conception clinic to aid these patients. Regarding the healthcare policy, there should be an increase in the infrastructure and the staffing of the pharmacovigilance office. There should be a framework for involving stakeholders in the creation of these alerts and messages. As for now, these communications are sent, but we're not sure if the recipient is understanding what is written or not. In addition to the pharmacovigilance needs to be publicly available by posting it in the MOH website and sending electronic emails to the healthcare professionals. And there should be documentation of the patient counseling regarding the valproate risks. There are multiple opportunities for future research, for example, to investigate which strategies that could improve the adherence to the recommendations of the pharmacovigilance. In addition to evaluating the impact of the pharmacovigilance recommendations on the patient outcome, as now the majority of the studies are evaluating the knowledge and the behavior change, which are surrogate to the patient outcomes. To conclude, as we all know that patient safety is not the sole responsibility of one organization, it requires the collaboration between the different key players, including the healthcare institutions, the patients, and the healthcare professionals. The communications would fail in terms of protecting the patients if they were not received and understood by the intended audience, and if there were no actions following these recommendations. And thank you very much.
Thank you, Dr. Amal, for your presentation. And if you guys have any question, you can follow up with Dr. Amal after. While we start setting up, maybe for the next presenter, if you can help me, Afrah, for the setting up for the slide. So our next presenter will continue on this section 2, clinical pharmacy, Dr. Abdurrahman al-Shaya. He's our first presenter from Saudi Arabia. He'll take us through the early management of traumatic brain injury. Now, Dr. Abdurrahman Shaya is an associate dean of clinical affairs, as well as an associate professor at the University College, at the King Saud bin Abdulaziz for Health Sciences. He's also a critical care consultant at the King Abdulaziz Medical Center or Medical City in Riyadh, Saudi Arabia. Dr. Shaya completed his Bachelor in Pharmaceutical Sciences from the King Saud University and a Doctor of Pharmacy degree at the University of Tennessee for Health Sciences Center, and that was completed in 2017. Then he later completed his PGY1 residency from Brigham and Women Hospital, as well as he completed a residency in second year for critical care pharmacotherapy from the same hospital, both of which are ISHP affiliated. Both of them are Harvard affiliated as well in Massachusetts, Boston, in US. Dr. Abdurrahman is a board certified pharmacotherapy as well as board certified in critical care pharmacotherapy. What's really not written in these words is that Dr. Abdurrahman is a really close friend as well as a colleague. We both did our residencies together, and I would say he's the best person to debate you with aminoglycoside dosing at 4 a.m. in the morning. So help me welcome Dr. Abdurrahman to the podium, please.
Can I use the portable?
You can use the portable, yeah. I'll turn off this one for you.
Bismillah. Okay, nice. Sorry, I don't like sitting behind the podium that much, so how is the voice now? Good? I have them on my Gmail, so.
Oh, okay. Do you want to log into your Gmail then?
Yeah, sure.
And we're back with March Sadness, and the team is looking down the barrel of a 4:55 meeting.
All right, alhamdulillah, we made it. طيب. So, all right, if I may, just want to double check, how many Arabic speakers and non-Arabic speakers here? How many non-Arabic speakers here? Non-Arabic first. 1, 2, 3, 4. Okay, good. So just wanted to double check how, if we can switch languages or what language should they just probably use. So first of all, thank you so much, Dr. Imad. Thank you so much for the committee for having me to talk about a near and dear topic to my heart. So I don't want to take much of your time. I know it's almost lunch break, so I will try to make this quick, although I have roughly around 60 slides, so I'll just have to fly in over these quite quickly. So my name is Abdurrahman Al-Shaya. I'm an associate professor and also associate dean and a consultant critical care pharmacist. And I'm particularly also covering a neurotrauma unit at my current institution. So what I'm going to present to you now is something that I usually do in my day-to-day practice as a clinical pharmacist in Saudi Arabia. So a couple of objectives that we're going to talk about. I cannot talk about traumatic brain injury without talking about trauma as an overall prospect. And without having this, as part of our thought processes, it's going to be really hard to understand TBI without understanding the overall prospect of trauma as a whole. So when we think about trauma, it's not just a one side of things that we usually think about. It can be just as simple as maybe a trauma that can happen from a motor vehicle accidents. It can also be happening because of homicides or things like that. There are also other reasons that can be related to military or fall or any other reasons. So it doesn't have one reason for you to think about trauma on its own impact, and it doesn't have only a physical impact. As a matter of fact, the first time the phrase of PTSD was phrased was actually after military combat being developed. So I can simply say that for us as pharmacists, knowing trauma and knowing exactly the consequences of trauma is going to be very, very important, because depending on that, you're going to intervene with whatever is going to be available. So it was medication-wise, or if it was some sort of blood products that we usually manage also from our prospect as pharmacy as well. So it have this triad of human, energy, and also environment. So if we're thinking about the environment itself, if the environment was quite dirty and the location of the trauma was not as clean, so you happen to have, I would say, a sterile trauma. If you go to the OR and someone is already cutting your abdomen open, this is technically an induced trauma, but however, it's going to be sterile trauma. So therefore, you wouldn't be worried as much about the consequences of this particular trauma. Versus if you happen to go into combat and you're already going to military mode and you're trying to hide under the bushes and someone is already hitting you with something, then that particular environment is not as sterile. Therefore, you have to think about the consequences of whatever is going to be happening, if it was a antibiotic prophylaxis, maybe certain vaccines and things like that. So that's where it gets to be more pharmacy friendly, even though we don't talk about that quite much. And then also you have the human factor. We know that humans are not always the same. You have different defense mechanisms. Some would probably have immunosuppressive factors, and some would probably have a different mechanistic approach from pathophysiology standpoint. Maybe it can take you on a journey in which what is the likelihood of someone to die from trauma. As a matter of fact, there is a global trauma foundation that was looking into that, and they were saying that roughly around 1.2 million actually die daily almost, yearly from prospect of trauma. And if you look into the local prospect, the third leading of cause for death in the Middle East is actually trauma, secondary to cardiovascular and also cancer as well. And then amongst that, 50 to 60% actually die within the scene itself. So it's not as much as the hospital care, it's the pre-hospital care that we have to do better at. And that's where it gets to be quite tricky for us as pharmacists, because we don't usually have this much involvement in the pre-hospital care yet. I can challenge that by possibly introducing certain protocol that can help you to understand the process as a whole. So if we can break this down, one of the things that can be as consequences for trauma is hemorrhagic shock. And that's going to be basically the most effective things that to kind of handle when it comes to hemodynamic assessments. From that kind of point, you also happen to have an increasing chances of death. That can be related to TBI or itself or the consequences of TBI. I can take you on a journey here of the evidence that we currently have on the data regarding TBI. So this is actually a Kuwaiti study that looked into the Middle East, and then the incidence of the reported studies that was reported to tackle trauma or TBI specifically. And you can see that the highest countries that were having published studies regarding TBI was Saudi Arabia, Qatar, and also Emirates. Maybe Kuwait was not doing as much in that area. However, I think that there is also an area of improvement for everyone here, specifically if we're talking about the third cause of death in the Middle East as a whole. So previously, the thought process was there is two scenarios for people to die from trauma. The first one is that the accident happens and people die because they happen to have this hemorrhagic shock, loss of blood, and they just pass away. So the second thing is that if they did not die at the moment, they were probably gonna stay at the hospital for maybe a few weeks, and they're gonna develop maybe sepsis or some other consequences, and they're gonna die later on after a few weeks from that perspective. However, given that we have better control currently at the pre-hospital care with the advanced emergency medicine services, now we are moving more towards having a possibly what we know as a three-wave scenario. So in that case, maybe we can push the mortality to be saved a bit more, so we can probably do better at the admission date in the hospital. So we're going to talk about a few things, a few concepts. We're going to talk about the MAP, which is basically the cardiac output multiplied by the systemic vascular resistance, and also we're going to talk about ICP, CPP, and also cerebral blood perfusion, and the definition as you can see here in the slides. So just to give you a quick prospect, possibly once we talk about TBI, the first thing that comes to someone's mind is ICP management. However, I'm not going to talk about ICP management alone. I'm going to talk about the overall care for trauma patients. So this is going to be only partial talk that I'm going to cover at the moment. So what we know as far as this point goes is that CPP is going to be the MAP minus ICP. And you definitely usually want to keep the CPP as elevated as possible for you to ensure that there is enough perfusion towards your brain. So one of the modalities that sometimes we try to do is that for you to have this thought process of maybe potentially having this hemorrhagic shock is going to reduce your stroke volume, and therefore losing this stroke volume is possibly going to reduce your cardiac output as consequences, and therefore there is a risk of hypotension in the beginning, which might be reflective of also having a reflexive tachycardia and some of the other consequences as well. So the data that we currently are seeking for, at the moment is possibly to introduce norepinephrine to slightly elevate the MAP towards all the way to 80 to 85 in that range, just to make sure that we're already having enough ICP in the range of 20 to 25. And that's where the cutoff is going to be for that prospect. I'm a very story-led person. I like to make stories. I like to make connections through stories. So let me take you through a story. So we have here Mr. Ahmed. Mr. Ahmed happened to have a nice family. He wanted to drive his car. It's a very nice GMC car that he usually drives. And he drove his family, and accidentally he happened to have an accident. So the thought process now is, what can we do to Ahmed so we can make sure that he goes back to his family in a very nice scenario? And the thing that I would like to ask everyone here, I would like to ask you to see what kind of questions, what kind of variables that you're usually going to think to ask before you try to save Ahmed's life. Let's make this interactive. What do you want to ask? Anyone? Can we have mics here? It's a logical question. It's not, even if. Excellent. Right, yes. So you definitely want to see what's the cognitive status for Ahmed. Is he already, what's the GCS, what you call it? There is also assessment tools called GCS, right? So what's the cognitive status for Ahmed? Is he awake? Is he not awake? Is he able to move? Is he able to interact? Right? So it's not going to be the same. So what else? What factors would determine Ahmed's survival? Vital signs. Excellent, right? So you'd like to make sure what his status for blood pressure, what is his heart rate is going to be like, and some of the other scenarios. Excellent. What else? Here in the back left. I think most of you guys are students, right? If I can point. Sorry, I do that to my students as well, so I have to do it here. Yeah. Anyone? Any wild ideas? It's fine. There is no way to go wrong, literally. Cognition, possibly. It's kind of within the same exact cognitive value. So not to take much of your time, there are a couple of things for you to think about. Is this really a TBI, and how severe is the TBI, and a couple of other risk factors. So these are the things that you're usually going to consider. So what kind of things that usually happen? So you're going to ask yourself, is this a penetrating trauma, or is it a blunt trauma? So the difference is that penetrating trauma is, from its definition, it's penetrating. So there is a fracture, there is blood, there is something that is usually going to be seen in your naked eye, right? Versus blunt this is usually we're going to say, يمكن in Arabic, if I may, it's معم بنزيف الداخلي. This is literally what we know of. You cannot really see it, right? And therefore, the mortality rate for blunt trauma is higher because you cannot basically see it, right? Versus the penetrating trauma usually have better survival because you can actually recognize it, you can actually see it. The second one is that, is this only a head trauma, or is it also combined trauma? Is this only, there is head involvement, or maybe his leg has also been impacted as well? And then also the location of the trauma itself. We know that the brain pathophysiology is not the same. So being hit in the front is not going to be the same as being hit in the back. And also being hit in a certain direction, certain speed is not going to be the same as being, hit when you're, playing maybe with your friends and they're just hitting your head at a certain point. That's going to be slightly different, right? So you're going to also ask yourself about all of these particular questions. That's where we're going to go for the spinal involvement. Because we know that the CNS is not only brain, it's a brain and also a spinal cord involvement as well. And then also age is going to be an important factor. And why would age is going to be an important factor? Just thinking. Why does it matter? Why should we know, or why should it matter for me to know that maybe Mr. Ahmed, who's 70 years old, is gonna probably have a slightly different set of expectations and maybe consequences than maybe Selma, she's maybe 10 years old. So the consequences of this basically secondary to brain atrophy. As we grow old, naturally your brain is trying to get shrinkage, right? So this shrinkage format is gonna create kind of more space in your brain, and therefore your brain is gonna tolerate probably a slightly more blood in that size. So you wouldn't be getting into a lot of different consequences in that aspect. So the definition here is that not all TBIs are created the same, right? So we happen to have a certain classification cascades that's going to be from the Neurocritical Care Society, and it's going to be classified based on either GCS score or also the CT scan and some of the other ones. So just thinking about anatomy, as usually us as pharmacists are not usually anatomy oriented. However, in this particular scale, you're usually going to have some basic knowledge about brain anatomy. So you happen to have the starting points from the skull, and then you also happen to have the dura mater, and then also the arachnoid mater, and also you have the pia mater. In the Arabic also dictionary, sorry for non-Arabic speaker, but we also have our own definitions for that. And I usually like the Arabic definitions because I think you can relate to it. So the functions of these meninges is actually to protect our brain. You have a mushy tissue that is going to be lovely kept in a very spacious format within floating over a CSF space. So the idea of having your brain floating over this much of fluids is actually to save your brain from being hit every single time you're moving your head one way or another. So having all of these different meninges is also going to be quite important because this is going to later on give us an idea about what we call as compartments. So if you happen to have one space, it's going to be one compartment. If you're going to have more than one space, we're going to call it multi-compartmental issue. So in that particular scenario, we happen to have an epidural space, we happen to have a subdural space, we also happen to have a subarachnoid space, and then anything below the pia is going to be also called intracerebral hemorrhage or interparenchymal hemorrhage. And then beneath that, we also happen to have something we call it ventricles. I know Dr. Ahmed, he's going to talk also about heart, but we also happen to have heart ventricles within our brain as well. So unlike the heart, in which how many ventricles do we have? How many ventricles do we have in our heart? 2, right? So we can probably, the brain beats the heart in this, so we have 4 ventricles in our brain. And then the functions actually of the ventricles is actually to create CSF. We know that when someone goes and try to kind of get the tab for the spinal tab and the CSF kind of comes out, usually you happen to have consistent release from the ventricles for roughly between 500 to 700 cc's of CSF from this particular location. So if you don't have enough and proper drainage for the CSF from the ventricles is going to get swollen, and once it gets swollen, it's going to take another space from the brain, which is basically going to follow what we know of as the Monro-Kelly doctrine. So your brain is currently happen to be in closed space, right? And you have three different variables. You have bones, and within that bones, you have three different things. So you happen to have brain, which is going to fulfill 80% of this particular location, and 10% is going to be the CSF that's going to provide your brain with nutrients and also this caution kind of safety net. And also you happen to have blood that's going to provide your neurons with oxygen and all of that, right? So this is going to be the proper scenario. However, if you happen to have a bleed inside the blood, inside the brain, that means that this blood is not going to take 10% anymore. It's probably going to go into 20%, maybe 30%, and guess what? Where is it going to take it from? It's going to take it from the brain. So the brain size is not going to be 80% anymore, so it's going to be probably 70% and maybe 60%, which is going to be creating more pressure, right? So now maybe it's going to be a great time to talk about what is ICP and how can we monitor it. So from that particular standpoint, you happen to have multiple things that we can utilize. We don't usually see it unless you go to the ICU and see all of these different devices in which you're going to have an epidural, interparenchymal, subarachnoid, kind of intracranial pressure assessment tools that we can usually utilize. There's also something we call it EVD, and that also can be seen in the ICUs. And there is also a particular set for that, and there's also supposed to be trained nurses for this to be done in a very appropriate manner. I'm not quite sure how are we doing with the time? Just don't want to. We're good? Okay. So this is basically what we usually try to aim for, and just double check from a intracranial hypertension standpoint. So normally you happen to have this appropriate range between 5 to 10, and then usually the cutoff possibly is supposed to be set at more than 20, 22, in which you're going to have this thought process of that probably you have to treat that intracranial pressure in a very serious manner. And the causes for this increase in intracranial pressure is going to be all of the followings. However, we're talking about trauma here. So once we talk about trauma, I would like to highlight all of the possible causes here for trauma precisely. So within trauma, it's not just trauma. Trauma can also lead to a hemorrhagic stroke, which also can lead to hydrocephalus. And hydrocephalus is basically, do you remember when we talked about the ventricles just getting swollen? This is basically is hydrocephalus, right? And then we can also develop status epilepticus. You can also have hypercapnia and also hypoxemia and hyponatremia, which is usually roughly happening between 20 to 30% in patients with trauma. And you can also develop what we know of as a central fever, which is usually a non-infectious leading cause for fever. And the main cause of TBI, okay, so I'm told that they have 10 minutes, so I'll have to kind of fly over this quite quickly. So from the headache standpoint, this is usually going to be the main big thing that we have to notice. And from that perspective, the second major thing that you're going to see also within TBI, ICH, most of the brain stuff is going to be mostly headache and nausea and vomiting as a very important ones. What we usually get afraid of is not just that having ICP is bad, right? I usually like to ask my students, and so what? So if you happen to have ICPs that's going to be slightly higher, then what's going to happen? And the answer here is that we, as we just has talked about, so we happen to have this three different format in which you have 10%, 10%, and 80%. So this 80% is within enclosed space. And if you keep pushing the brain with this significant amount of blood flooding over this particular location, that means that there is a risk of something we call a hernia. And hernia in Arabic means fatk of tag. This is actually the Arabic translation for that. So basically, this particular thing is basically meaning that the brain itself is going to get outside of its pocket, which is going to lead to a brain death scenario, right? So this is what you're going to be afraid of. It's not just that you're afraid of the ICP. The ICP is just a tool for you to avoid having herniation on its own. So this is where it gets going to be quite tricky. And not all herniation are bad. Some of them are quite bad, and some of them can be managed appropriately. But your goal is hopefully to not have that as much as possible. So your therapeutic goal, hopefully, is to keep an ICP below 20, and also for CPP to be between 50 to 70 using norepinephrine or any of the other ones. So there are a bazillion kind of strategies to do and deal with these kind of scenarios. So let me just kind of go over a couple of these ones. So in addition to having a TBI, we had just also talked about the spinal involvement. So possibly also having a spinal involvement is going to be quite important for you to think about. So thinking about the functionality for certain neurons, and also how are they going to be innervating particular muscular systems. You also happen to have, if you happen to have also injuries to the C3 all the way to C5, in that particular case, these are the ones that are going to usually drive the innervation for your pulmonary system. So these patients at a higher likelihood of them needing possibly a higher level of sedation, or maybe not having a proper response to sedation if you're going to assess the follow-up scenario as well. And they also happen to have a slightly different responses, which is slightly weird. So in spinal shock or spinal cord injuries, the most commonly seen consequences is bradycardia, despite having hypotension. Because as we had just mentioned in the hemodynamics, usually when you have hypotension, for your body to correct that, you usually have reflexive tachycardia. However, with the spinal cord injuries, you're usually going to have a hypotension with non-corrected bradycardic scenario, which needs to be intervened immediately using norepinephrine or some of the other scenarios. Sometimes we also use phenylephrine. So that's going to be also another thing that we usually have to see. And then the best data that we have for spinal cord injuries is actually to aim for a MAP that's going to be more than 85 to 90 using norepinephrine. Some data suggests that using phenylephrine also is going to be important. However, both for TBI and spinal cord injuries, data supports not giving steroids. Do not give steroids for patients with spinal cord injuries or patients with traumatic brain injury as well, because data suggests that if you happen to do that, the risk of mortality is going to skyrocket as well. So there are a couple of other consequences that we can also see. There is a surgery we call craniotomy and craniotomy, basically just going to the brain, just taking some part of the skull out and just keeping this outside and just letting the brain breathe. Just give it more space for it to actually can expand. So you just get outside this enclosed location and try to save the patient as well. I highly recommend you to review this checklist. This is a very, very important checklist. I'm not going to go over it, but this is very, very essential for you if you would like to save patients' life who are coming with a traumatic brain injury. So in addition to ICP management and all of that, you also have to think about, again, what's the source of trauma and how can you also save patients with trauma as well. So these patients can come also with an open wound fracture. An open wound fracture means possibly a likelihood of injury that's going to be, a likelihood of infection that's going to be slightly happening there. So there is also a classification, we call it Gastio classification. And then based on the Gastio classification, the data that we have so far is that if we happen to have one or two, possibly you can use Vaseline for these patients. However, moving forward, we can probably use a minor exercise in these particular cases. And then we can probably kind of tailor it for a very short period of time. And then in addition to that, we also have big rules as pharmacists in vaccines, and we rarely push for certain vaccines. And these are one of the quite important scenarios in which we have to push for vaccines. So Tdap for patients with trauma is going to be very important. And in certain scenarios, it's not going to be the only one. You have also to add IVIG in a very appropriate manner. So the priorities that you're going to have, definitely assessments that's going to be starting with ABCs, airway, breathing, and then circulation, and then moving forward all the way towards having a proper scenario to treat your patient's case. And then just to kind of go quickly for sedation purposes, there are data for multiple different sedatives, including propofol, dexmedetomidine, and remifentanil as well. But propofol is usually the most common, the most common one that we usually are currently using. So there are so many hyperosmolar therapy that we can use to treat intracranial hypertension, including mannitol, hypertonic saline glycerol, sorbitol, or urea. And then if you think about the content of each one of them, we happen to have mannitol in different concentrations and also hypertonic saline in different concentration, 3%, 7.5%, 23.4%, and some institutions actually they use their own percentages. You can find 2%, 5% based on whatever institution availability is going to be assessed. Talking about mannitol has great data. Yet, it's not going to be your first-line therapy based on the most recent guidelines, though. So you have the range of dosing between 1 to 2 grams per kilogram per dose. And then in that particular case, basically mannitol is a gigantic glucose mimics. So it's going to induce basically a sort of hyperosmolar pressure that's going to be pulling the fluids from the brain towards the blood and then having this diuretic effect to relieve some of the pressure as well. However, there are a couple of consequences of using mannitol, so it's not supposed to be recommended to be used for patients with CKD or AKI. And also, if you happen to have the right to handle mannitol, if you keep it outside within the fridge or outside in the room temperature, sometimes it can form crystallization. And then from that aspect, if you happen to be on a trauma bay, in an area that has usually more trauma than others, there is a recommendation in which to have this already prepared in a warm bath to make sure that there is no formation of crystallization or anything else. So that's the dosing here. So it's just start from 0.25 to 1, and there's also data to go all the way to 2 grams per kg. And these are the consequences for mannitol as well. The hypertonic saline also happen to have better data that we know of so far. However, there is so many safety points that we have to be very curious about, and we have to make sure that we're not really going beyond the recommendations for it so far. So 23.4%, according to the ICMP, it has to be labeled as concentrated and not hypertonic, and it's supposed to be kept within the pharmacy and not to be sent to the floor. However, if you happen to have 3%, according to your nurse's familiarity, sometimes you can keep it in the floor, and it depends on your safety officers. I've heard our previous doctor, she was talking about the safety and pharmacovigilance perspective. This is usually going to be an institutional decision. It's not going to be something that's going to be done singularly as a pharmacist. So these are the recommended dosing is supposed to be given. Usually the doses we're going to have currently is recommend the boluses over the continuous infusion, because the boluses have this biphasic distribution. So you just create pressure, pulling the volume out, and then excreting the volume, and then also phasing out for 6 hours, and then you're giving another dose. So you just kind of keep pushing with this biphasic distribution format. However, it can lead to certain consequences. We're playing with sodium, so the risk of central point time melanolysis is going to be there, maybe risk of phlebitis also with the concentration as well. So I highly recommend you to follow the safety standpoints and monitoring parameters and availability as well to assess your patient's availability. One very important factor is that the monitoring parameter for mannitol is going to be the blood osmolality. So your goal is usually between 300 to 320 for patients. However, making sure that you are also understanding your patient. So from a sodium standpoint, if you happen to have patients with a baseline sodium of 125 and 130, your goal is actually basically to only increase it by 10 to 15%. Sorry, 10 to 15 millimoles per liter. So if patients came in with 130, your goal is to keep these patients between 140 to 145. So not all patients has to be kept between 150 to 155. This is not a common, it's a common terminology that's been running around, but if you look into the recent guidelines, just increase it from the baseline by 10 to 15 millimoles per deciliter. So these are a couple of the consequences that we just talked about, and then also there is also this great tier mechanism from the Neurocritical Care Society in which you have tier 0, tier 1, and tier 2, and tier 3 as well. Then also you have to correct coagulopathy if you happen to have that. So there are multiple data of using tranexamic acid, and there is also more data about using possibly PCC, maybe some of the other blood factors in that. The data so far, according to the TEC trial, the CRASH 3 trial, and some of the other ones is not very promising. However, you can maybe use it in patients with mild to moderate, and possibly if you can set a recommendations for that, it's going to be the ideal scenario. The second or the third major topic is also seizure prophylaxis. The data that we know of so far, based on the Trauma Foundation recommendation, is possibly using seizure prophylaxis for patients with severe TBI is going to be highly recommended. So using phenytoin or levetiracetam equally kind of have acceptable data. At our institution, we're using more levetiracetam because it's going to be slightly having less drug interactions risk and some of the other consequences as well. So try to avoid using these interventions. try to avoid using hypothermia. This is not going to be having a lot of promising consequences based on most recent meta-analyses. Do not use steroids, as we had just mentioned. And also, if you don't need prophylactic hyperventilation, please do not use prophylactic hyperventilation in your patients. So this is the summary that I have. Sorry if I'm going over time, but this is the last slide that I have currently. Just basically summarize the guidelines that we create at our institution. Sorry. Oh, that's a trauma. Okay. All right. So this is the slide that we're going to have. These are my references, and thank you so much for listening.
All right, thank you guys for listening. So I'm having a traumatic brain injury right now. I'm kind of hungry. I guess you all are. So we'll take a 30 minutes of break, and then we'll resume after. Thank you.
Saudi Arabia. And also he's a cardiovascular pharmacotherapy specialist in King Saud University Medical Center. And he's currently served as the chairman of the Pharmacy Scientific Council at Saudi Commission for Health Specialties and a board member in Saudi Society of Clinical Pharmacy. Dr. Ahmed has received his bachelor degree in the pharmaceutical sciences from KSU and PharmD degree from MCBHS University. He completed his BGY1 and BGY2 in cardiovascular pharmacotherapy at Brigham and Women Hospital. We welcome you, doctor, and you may step up.
Assalamu alaikum. Well, I would like to start by thanking Kuwait University for the invitation and the scientific committee and organization committee for the outstanding Congress. Starting off with our updates in heart failure, I just have a quick question. How many are practitioners in cardiology practice? You have only one practitioner. Okay. How many like cardiology? Okay. How many like critical care? Okay, good. So actually the reason why I chose to discuss heart failure is one of the things that is really paradoxical. So from my opinion, it's the easiest disease state to treat on paper. So patient has a lot of fluid, give him diuretic. Patient has hypoperfusion, give inotropes. It's pretty easy. But when it comes to real life, patients never go through the scenario. So starting today, we're gonna have a quick overview of the guidelines, American guidelines. So we have an update there. And then I'm gonna show you quickly the updates from the European Society. The reason why we're gonna do this, just to show you that there is very strong alignment between both of them. So regardless you go with the American versus the European, you're in a good shape. And then we're gonna have quick take-home messages. If you think about the management of heart failure, people usually talk about medications. So they talk about ACE inhibitors, talks about ARB, talks about all these pharmacological groups. To me, I'd like to go the opposite side. So when you think about heart failure, you're gonna hear this terminology, guideline-directed medical therapy. Do you know which medications is considered guideline-directed medical therapies and which are not? People sometimes, maybe students usually, they try to memorize it. But how to figure it out is pretty easy. I'm just gonna take you through, instead of starting with these medications, I'm gonna take you through the history of it. But before going to the history, I wanna share with you some data that's a little bit concerning, to be honest. So the data in front of you right now, this is Gulf data. So it's Saudi, Kuwait, Emirates, Oman, all of the Gulf area. You can look at the prevalence. The prevalence is 1 to 2%. The number looks low, but look at the number. Instead of percent, look at the number. We're talking about 3.7 million patients in our regions having heart failure. On average, each patient is taking 10 medications. So we're talking about, if let's say one medication is $1, we're talking about $37.5 million per patient is happening right now. The second point is a really devastating point. If you look at the prevalence of it, who are the demographics of these patients? In our region, when patients get diagnosed with heart failure, they are 10 years younger than their similar patients in Europe. So in Europe, usually they start having this heart failure around 60s, you know, as an elderly. For us, they start pretty early, sometimes early 50s and 40s. And this is very concerning. It has to do a lot with the lifestyle that we have and some genetics as well. If you look at the study that just showed 1 year mortality, you can see that there is 9% all-cause mortality. The majority of it is because of cardiovascular diseases. And the emergency room visits is almost 50%. Okay, so now let's go to the story of it. So if you go a little bit back in the history of heart failure and how did people think about it, it was pretty simple. When you think about heart failure, it means that the ventricles are failing to supply blood to the body, low cardiac output, period. So anything that causes the contraction of the heart to go down, anything that causes the blood flow of the heart to be reduced, this is technically making heart failure worse. That's why if you go back 35 years plus, you're gonna find that beta blockers and calcium channel blockers had black box warning. Do not use them in heart failure. But what I just said now is contradicting what's happening. People are now using beta blockers for survival, for mortality benefit. So what changed? The reason why they understood more the pathophysiology. And that's a slide in front of you right now. They did more studies and said, okay, what's happening? Why the body is having heart failure? The simple way to do it is it's a normal mechanism. Each one of us is going through whatever you're seeing in the screen right now. When you have Chinese food with high sodium in it, your body does not have all this fluid overload. What's happening? Pregnant women, they have a lot of fluid, so why don't they have heart failure? Because of the systems you're going to see right now. So they figured out there's two things. The heart is feeling or assessing that I don't have enough blood going to the organs, so what's going on? And hypotheses start coming up. It might think, well, I'm not pumping strong enough, so it goes stronger. Sympathetic nervous system goes up. I'm not pumping fast enough, sympathetic nervous system again goes up. Sometimes you think, oh, maybe the vessels are dilated. Can I constrict it? And that's what it does by the RAS system. So the angiotensin goes up. Sometimes it says, you know what, maybe I don't have enough fluid. So the RAS system gets activated, water retention. That's why when they come to the emergency, the number one symptoms they have is fluid overload. And then lastly, they think, okay, maybe I'm really constricting and contracting, but the muscles has to be bigger. The muscles do not get bigger without hormones. So that's why we have aldosterone in the RAS system going up, and that's how we block it with spironolactone. So the ultimate goal, if going back to my question about GDMT, guideline-directed medical therapies, any medication that affects these two pathways is a GDMT. You don't have to memorize it. So GDMT is a medication that affects one of these two pathways, and by doing this, reduces mortality and reduces hospitalization. But the reason why I'm starting this because there is even more to know about this. Someone can ask me, the SGLT2 inhibitors, the antidiabetics, now they are used in heart failure, but where do they fit in this? The pathophysiology of heart failure is becoming, the more we know it, it's more complicated. And again, you can see in the top right of the slide, we're talking about the natriuretic peptides. Natriuretic peptides, it's something that is very interesting to understand, because if you see very high BMP in your body, that means they have bad prognosis. But is it a bad marker? The answer is no. It's amazing. The BMP is basically sensors in the atrium and the ventricles. They are overlapped. Once the ventricle stretches, that means that there is a lot of fluid. At that time, the heart does not communicate to the organs through the brains. It wants to talk to the organs right away. So how can the heart talk to the kidneys? They don't have a common language. So instead of saying get rid of the water, it says get rid of the sodium by BMP. So BMP telling the kidneys get out some sodium and water leaves. And that's what's happening. So that's a BMP. What's the benefit about this? This is one of the paradigms in heart failure, the Entresto. So sacubitril valsartan is a combination of sacubitril valsartan and as a combination to get rid of neprilysin. Neprilysin decreases the BMP, so to have it higher in the body, it's as if giving a natural diuretic without side effects. But the problem is, once you do this, the angiotensin 2 is also broken down by the neprilysin. So if you block neprilysin, angiotensin 2 is going to increase too and going to cause you problems. That's why you never give sacubitril by itself. You have to give ARB with it, which is valsartan. The reason why valsartan, because valsartan and sacubitril are produced by the same company, both of them are Novartis. So there is no preference between the ARBs. So this is basically the story. But before we go to the guidelines, we need to agree on one thing. What is the treatment of heart failure? Anyone knows the treatment of heart failure? What is it? Transplant. So we need to keep in mind that the only treatment for heart failure is transplant. We manage symptoms. We slow the progression as much as we can by blocking these neurohormonal systems, but we cannot treat it. It's transplant. And based on the transplant, I just want to reply to Dr. Abdurrahman when he took the ventricles from the heart and he said the brain has ventricles. I would like to let you know as well that the heart has 12,000, 20,000 neurons in the heart. And the way they figured out this is by transplant. When they did heart transplant, they figured out people has memories that they never lived before. They had memories from the patients they took the heart from. So the heart does have neurons in it. Sometimes when you smell something or you have a feeling in your heart, there's a huge connection between the neurons and what we call have you heard about the broken heart syndrome? Yeah, it's about the feelings. The feelings can really, technically, feeling sad, change the shape of the heart to be like a vase, and this can literally kill the patient. It causes cardiac arrest, usually happens with females because of emotions. It's not something that has medications for it, but this is what happens. So we do have neurons as well, Dr. Abdurrahman. Okay, so American guidelines. These are just the colors. We're going to be using it throughout the recommendations. I'm going to show you a brief of what is new in the recommendations of the American guidelines. When you think about heart failure, you think about the most common one that you see so far is stage C. So stage C is the common one with heart failure with reduced ejection fraction. Stage A and B, these are pre-heart failure, so they don't have heart failure. To simplify things, this is on the slide. It tells you if the patient has hypertension, treat hypertension. Is there a specific medication you can use? Not necessarily. You can go to the hypertension guidelines. But when it comes to diabetes, is there something preferred? The answer is yes. We use SGLT2 inhibitors, and this is dapagliflozin or empagliflozin. Both they have guidelines, recommendations. Cardiovascular disease, whatever they have, you have to fix it. So this is the at risk for heart failure. If you look at the other side, the stage B, you're going to find that we're talking about ejection fraction that's pretty low. So how can I differentiate between patient having heart failure and pre-heart failure? The answer is about the symptoms. So if there's no symptoms, then we're fine. You can treat it as pre-heart failure. You can look at the stage B. You're going to see the medications pretty much similar to the GDMT. ACE inhibitors, beta blockers, and all these things. So take-home message from this slide, they went in the guidelines to the details of how to treat your patients based on what they have to prevent the progression of heart failure. Now this is stage C and D. Stage C is the most common one, so patients start having symptoms. There is changes in the anatomy of the heart. D is usually the end stage of it, so we go on inotropes, stuff like that. We commonly say ACE inhibitors, we say ARB. Can you see it on the slide? ACE inhibitors and ARB. So do you see the first column on your left? These are the things that the patient must be on it, guideline-directed medical therapy. They started with ARNI, which is Entresto, because they found out that the benefit out of it is bigger than ACE and ARB. If they cannot take it, start ACE and ARB. In the previous guidelines, they did not. They said ACE, ARB, or ARNI, all of them, because it was expensive, but now there is no justification only to have 3 of them together, so the preference is with the ARNI. Beta blockers, for sure, to block the sympathetic nervous system. The MRA, which is spironolactone, to block the aldosterone, so it's a RAS system. SGLT2 inhibitors for diabetic or non-diabetic patients. Because I told you now everyone takes this, right? But this is a diabetic medication. The answer is regardless. So your patient has diabetes, your patient does not have diabetes, give SGLT2 inhibitors, and then as needed diuretics. One of the advantages of the SGLT2 inhibitors is it has this diuretic effect. And one of the biggest problems we have in heart failure management is how physicians look at diuretics. They think it's, Lasix, everyone knows Lasix. Lasix is a disaster. Lasix increase mortality, but people do not do this. They do not know this. They say, okay, I'm gonna give you a minimum dose of Lasix. The reason why, it's just a prophylaxis. Do not do this. This is increased mortality. So now with this introduction of the SGLT2 inhibitors is gonna help with this effect. Again, if you look at what's after this, African Americans, we give them hydralazine and these things. I'm not gonna go to the rest of it because the rest of it is devices, so it's outside of the scope of the pharmacy practice. This is what I was talking about. You cannot talk about therapy right now without touching base on the value-based. So you are treating the patient, you are going with the guidelines, but what value are you giving? Are you improving the life of the patient, the quality of life? Are you reducing hospitalization? Are you reducing mortality? And this is something you have to keep in mind. All the ones in green shows high value. So that's why if you look at the second box, it tells you if you have Entresto or ARNI, please use it. It has high value. If not, then use ACE or ARB, and that's how you move in terms of using the other agents. Because of the lack of evidence, we don't have a lot of evidence. You can see SGLT2 inhibitors still has intermediate value because of the price. So you can look at what are you helping versus how much you're paying. Okay. I just want to highlight two things on this slide. These are other things that we use in addition to the GDMT. It's an add-ons. Please, digoxin, my take-home message, this is very, very important. We did this in the US, and we almost killed a lot of patients. Once the guidelines or the evidence showed that digoxin does not really work as much. We do not need it. It needs a lot of adjustments and stuff like that. But the problem is, once we started doing this, we stopped. Patient is already taking digoxin. We start stopping it. Mortality increased, hospitalization increased, and we didn't know what's happening. It turns out that the patients who are on digoxin, their body get dependent on it. So we call digitization. So they are dependent on it. Do not stop it. Keep it, but what do? You give very minimum dose. So your target is 0.5 as a plasma concentration instead of the usual 1 or 0.9. Do not stop it. Do not stop it. Do not stop it, please, because this is really serious, and people try to switch to the new agents. Just last month, we lost 3 patients because of the same way. They switched the patient. They stopped digoxin, they started Entresto, and the patient went to a very bad side effects, and they did not make it. The second one, the PUFA, this is the fish oil. It has some evidence to be used in heart failure. Again, you can use it, and it's 1 gram per day. There is good evidence so far, but still we need more data on it. That's why the data is moderate. Okay. This is a new concept, the people who are between 40 and 50 ejection fraction. Who are these? So reduced ejection fraction, less than 40, preserved above 50. So what happens in between? In between, what do we give? SGLT2 inhibitors. Okay, so the antidiabetics, that's what we give. Other things, it depends. So we don't have good data. ACE, ARB, beta blockers. So if a patient comes to you and you are in doubt, just give dapagliflozin, empagliflozin. And same thing for heart failure with preserved ejection fraction. I would like to ask a question just to see the perception of you guys. So we have 2 types of heart failure, right? We have heart failure with reduced ejection fraction, which is less than 40, and we have heart failure with preserved ejection fraction above 50. Which one is worse, from your opinion? Who says reduced ejection fraction is worse? Okay, we have a couple of hands. Who says preserved ejection fraction is worse? Okay, we have 50-50. So the answer is preserved ejection fraction. The reason why, there is no therapy. Preserved ejection fraction, we don't have. Everything I'm showing you right now, this is on reduced ejection fraction. Preserved, we usually go with symptoms. We have some data on spironolactone, some data on Entresto, and SGLT2 inhibitors. But other than that, it's even weak data. So although it sounds better, preserved ejection fraction, but it's worse. Okay. This is very interesting outcome as well, meta-analysis. If you practice and you see patients with heart failure, we say, do not drink a lot of fluid, restrict sodium. Is this true? Is it helping? And that was one meta-analysis. It's not that big. It was on 6 clinical trials. And what they found out that restricting fluid intake did not change in terms of mortality and rehospitalization for patients. That's why even in the IV diuretic dose, it did not change. So it's a lifestyle modification. That's what really matters. It's not about the fluid. Sometimes you really need the fluid because patients might have fluid overload. They seem like it, but they are dehydrated because the fluid is not inside the vessels. It's outside. So the body is not being appropriately perfused, and you are restricting the water, you make things even worse. And you get the patient to renal injury and all these stuff. So that's why the recommendation about lowering the fluid intake is still uncertain. You have to be per patient preference and what you want to get out of it. So this is pretty new recommendation. It reminds me of the same recommendations that happened with the dyslipidemia guidelines. If you look at the previous guidelines, they said if you have dyslipidemia or hypercholesterolemia, they said do not eat shrimps, right? You have to minimize your oil intake without specification. The answer is it's not true. They did not find any correlation between the plasma concentration of the cholesterol and the amount of food you are eating. That's why, if that was true, other agents that prevent the absorption of cholesterol would have the best outcomes. Although statins does because it prevents the production of the cholesterol from the liver. So if you go to the guidelines right now, you're not going to see in it, do not eat eggs, you're not going to find it there. Do not eat shrimps, you're not going to find it there. So we have some old conceptions that it has been revisited and can be revisited through research currently. Okay. Inotropes, take-home message, they kill patients. Okay. So inotrope, only use it if it's a bridge therapy. So if the patient is going to have a device, give them some inotropes until you get the device. If the patient is not going to make it, and you know they're going to pass away in a couple of months or so, they have the right to have a good life until it stops. So palliative, you can use it. Other than that, inotropes kill. Can you send patient home an inotrope? Yes, no? Absolutely, yes, you can. There are a cassette, the patient gets it, and it's like continuous infusion. Usually we use Mylinon for this, but again, it increased mortality. This is huge. I'm not sure in your practice, but again, I know many of you practice in Boston, and especially in the Brigham. There's a residency program called Transition of Care, residency program. And this is basically how transition of care is where the patient moves from one setting to another, from the emergency to the ward, from the ward to the ICU, from the ICU to the ward again, from the ward to the home. All these steps are point of care. Data shows that it has mortality benefit. So if you do appropriate point of care and transition of care, it's as if you're giving good dose of beta blockers, as if you're giving a good dose of ACE inhibitors. It is as important. The other thing is, do not delay. Please see the patient within 7 days. Because when the patient is in the hospital, you control what they're eating. You control what they're doing. But once they go home, they're living their normal life. Do not delay it. Is there harm after that? Yes, there is. You have to see the patient within 7 to 10 days maximum to optimize all the medications. Okay. Again, this is the way how to deal with heart failure patients. It's basically depending on what do they have. They have hypertension, fix hypertension. Diabetes, give SGLT2 inhibitors. And this is how things go about this. For AFib patients, AFib is highly linked with heart failure. Heart failure causes AFib, AFib causes heart failure. So you have to be very careful about this. Some physicians, what they're gonna say is, just in case, I'm gonna give the patient chronic anticoagulation. It's acceptable, although it doesn't have high grade of evidence, but we can use it. Because sometimes heart failure, they tend not to only have a clot in the atrium, they tend to have a clot in the ventricle as well, so you want to prevent that. Which agent should you use? The direct oral anticoagulants are preferred over warfarin. Okay. Iron deficiency, it's one of the biggest things that we take care of, regardless if the patient has anemia or not. The problem is not with the iron, the problem is with the erythropoietin, because I'm not gonna go to the details of this, but if you increase the hemoglobin of the patient pretty quickly in heart failure patients, you increase the risk of mortality. People think, okay, good, let's go up with the hemoglobin. No, it does not work this way, okay? And this is the introduction of the IV iron. It gives pretty much good outcomes. Sleep disorders is very important. Sleep is highly linked to the cardiovascular disease, so anyone in this room, students and faculty, please take care of your sleep. Like reduction of sleep completely has direct impact on your cardiovascular health, direct impact. Management of diabetes, again, who's the superstar right now? SGLT2 inhibitors, okay? Empagliflozin and dapagliflozin. AFib, I just mentioned that for you guys. Cardio-oncology is important, so which medication we usually use? ACE, the classics, the old one. ACE are beta blockers. This is patients who are taking oncological medications and they have cardiovascular toxicity or risk of cardiovascular toxicity. That's how it works. Going a little bit, we talked about value-based, and now this is another way we should, it's very interesting what they figured out. KPIs, key performance measures or indicators? They found that if you are using performance measures in your heart failure management, again, it's as if you are giving a correct dose of beta blocker and ACE inhibitors. It improves mortality. So keeping an eye, okay, who's in the team? In the United States, in Brigham and Women's Hospital, for example, the insurance company would not pay for the stay if the discharge is not signed by a pharmacist, a physician, and a nurse. Anyone missing, they're not gonna sign it, okay? So having this in your daily practice in the policies, performance measures, is as important as giving the patient ACE, beta blocker, ARNI, or SGLT2 inhibitors. Goals of care, we mentioned that before. Quality of life, this is very important. And you have to discuss things with patients. They need to understand, as we mentioned in the beginning of our talk, the only treatment for heart failure is transplant. We are managing it. So how the progress is going, what we're gonna do in between, all these things. I had a couple of slides, but I just removed them this morning. It was about how to deal with heart failure and pregnancy. It's a huge thing. So she has heart failure and she wants to become pregnant. What should you do with this? But maybe it's a talk for a different day because this is, I thought it was going to have a lot of discussion. I was talking with Dr. Fatma and other colleagues right now about research. I think in the Gulf area, we do need research. We are using, today, I was really upset because what data did I show you? American, European. Technically, anyone in this room can stop me and say, does not apply to us. Do you have evidence? My answer would be, you're correct, I don't have evidence. We need data, we need information. Yet, even the current American and European guidelines, if you look at all these are gaps in the literature that we can fill in. So from this place, I would like to, since we are in the Gulf area, we'd like to have research on this. We are spending a lot of money on this. We have a lot of patients, and literally, we are just doing a surrogate endpoint. We're taking what happened in Europeans and Americans and try to believe that this happens to us. So this is really unhealthy. We have the baseline to do our own research. I put this slide for you to see how much we can do. Just by copying this slide, you can at least have 20, 25, it's not even clinical trials, it's like a combination between economics and retrospective. As I promised you, this is the European guidelines. The only reason I want to show you is for the first time, there's 100% alignment. So you're going to see that the definition now is the same. So reduced ejection fraction less than 40, in between 41 to 49, and preserved above 50. Using the SGLT2 inhibitors, again, pumping up again. And they went specific. They didn't say SGLT2 inhibitors. They said dapagliflozin, empagliflozin, based on the studies. Again, same thing. When we talked about patients with preserved ejection fraction, we talked about patients with mildly reduced. We had only one option that has strongest data, SGLT2 inhibitors. It's interesting to have diabetic medications overseeing the market. This is the same thing, preserved SGLT2 inhibitors all the way. And the last recommendation is the same thing we talked about. Please, please, please see your patients within 7 to 10 days maximum. The Europeans laid back a little bit. They give you up to 6 weeks, but multiple visits. So they put an S here. And the American, they said, no, 7 days, 10 days, you need to see your patients. This is my last slide, take-home messages. As we said, SGLT2 inhibitors, they are part of the GDMT, regardless if the patient has diabetes or not. Please go ahead and do it. Digoxin, if the patient is taking chronically, please do not stop it. Don't kill the patient. Instead, you just lower the dose. SGLT2 inhibitors, whenever you think about preserved ejection fraction, mild to moderate, anything not less than 40, your number one way to go is SGLT2 inhibitors. See the patient within 7 to 10 days. And the last thing is, this is a little bit administration, but adding value measurements and improvement of quality. This is as important and as good as beta blockers, ARBs, and SGLT inhibitors. By that, I would like to thank you. Hopefully I'm on time. And thank you so much.
Thank you, Dr. Ahmed, for this very informative presentation. And now we can take questions for both doctors, maybe after like Dr. Thamer's lecture. I would like now to introduce Dr. Thamer, who is an associate professor at the Clinical Pharmacy Department, College of Pharmacy at King Saud University. And he's also a consultant infectious disease pharmacist at University Hospital. Dr. Thamer currently is serving as a chairman of the Clinical Pharmacy Department, and he's a director of infectious disease pharmacy residency program. With now, Dr. Thamer, I would like to welcome you for this talk.
بسم الله الرحمن الرحيم الحمد لله رب العالمين والصلاة والسلام على رسول الله. مساكم الله بالخير جميعا. في البداية أحب أشكر اللجنتين التنظيمية والعلمية والقائمين على هذا المؤتمر على جهودهم الكبيرة، خص بالذكر دكتورة فاطمة وأيضا دكتورة أفراح على كل جهودهم حقيقة الكبيرة ومتابعتهم لنا من شهور سابقة. سعيد جدا أنا وزملائي الكرام أننا نزور دولة الكويت، البلد الحبيب والقريب لقلوبنا واللي لطالما تطلعنا حقيقة أن نزوره منذ الصغر. شكرا على هذه الاستضافة الكريمة، على هذا الكرم الغير مستغرب، ويعطيكم العافية. So today, just as Dr. Fatima said, I'm infectious disease specialist. So I will be talking about infectious disease topic, which is actually very important nowadays, especially with the increasing rate of antimicrobial resistance, not just in the Gulf region, but also globally. So I have no real or potential conflicts of interest related to the subject matter in this presentation. So as you all know, that antimicrobial resistance increases and causes millions of deaths globally every year. And gram-negative resistance in particular poses a significant challenge in healthcare system. We have 6 main gram-negative resistant pathogens, and just for the sake of the time, I will be focusing on the first 2 of them, the carbapenem-resistant Enterobacterales and difficult-to-treat resistant Pseudomonas. So my objectives for today are to illustrate the prevalence of both pathogens, to describe the resistance mechanism briefly, and I will spend most of the time in the pharmacotherapy management, and especially the update in the 2013 IDSA guideline. So how serious CRE and drug-resistant Pseudomonas are? So the CDC updated their 2013 list of antibiotic-resistant threat after more data sources provide a complete estimate of their impact on the human health in the US. And now we have the 2019 CDC antibiotic resistance threat. As you can see, carbapenem-resistant Enterobacterales is considered a serious threat, whereas multi-drug-resistant Pseudomonas is considered an urgent threat, or it's vice versa. So in the 2017, the WHO published a list of microbes, almost 12 microbes, in which new antibiotics are urgently needed. And as you can see, carbapenem-resistant Pseudomonas and CRE are among the top priorities. So in the first half of my talk, I will be talking about carbapenem-resistant Enterobacterales. And whenever I say CRE, I'm referring here to carbapenem-resistant Enterobacterales. So simply, what is carbapenem-resistant Enterobacterales? So basically, they are Enterobacterales resistant to at least one carbapenem. So it could be imipenem or meropenem, or producing carbapenemase enzyme. So all CREs could be carbapenemase producer, meaning they have the gene that produce carbapenemase. And it could be KBC, which is very predominant in the United States, or OXA48, which is very predominant in the Gulf region, like in the Kingdom or in Kuwait. However, there is a substantial number of non-carbapenemase producers, and this is usually selected after the use of carbapenem. So we may get those pathogens emerged after the use of routine or frequent use of carbapenem for ESBL infections, for example, especially if we are using suboptimal dosing or there is incomplete source control. Looking at the Gulf region in terms of epidemiology of CRE, this data was published by the WHO, and they quantify the number of microorganisms among different gram-negative pathogens. And here I'm focusing only on carbapenem-resistant Enterobacterales, and as you can see, the rate in the Kingdom of Saudi Arabia of CRE is 15%, whereas it's lower in Kuwait, it's 5%. So you do better than us in terms of the CRE rate in Kuwait versus the Kingdom. So this figure illustrates the antimicrobial resistance in various regions in the Kingdom of Saudi Arabia. We published this study a few years ago, and we basically quantify the microorganisms among the resistant pathogen in a various region in the Kingdom of Saudi Arabia. Specifically talking about the CRE, among 12,000 pathogen published in a various region in the kingdom, the rate of CRE reached about 15%, which is comparable to the WHO report. So in this table, I'm just explaining briefly the beta-lactamases. I just mentioned that CRE could be carbapenemase or non-carbapenemase. Here, beta-lactamase, one of which is carbapenemase. So we have 4 different types of carbapenemase, or for beta-lactamases, A, B, C, and D. And I listed B at the end because it has distinct feature as it has a zinc ion. However, we may have serine beta-lactamases, such as narrow spectrum, the ESBL, or could be wider spectrum, such as KBC or OXA48, which inactivate carbapenem. NDM as well, we have IMP, VIM, and NDM, which inactivate all beta-lactam, including also the novel beta-lactam, but luckily we have aztreonam still active against it. The key from this slide is there is no single agent, even in the novel agents, that can inhibit all beta-lactamases. Here are the 5 beta-lactamases that we have in, I would say globally, KBC, OXA48, and the 3 metallo-beta-lactamases. It's interesting to see the global distribution of these carbapenemases. As you can see, KBC is the most predominant one in the North and South America, whereas OXA48 is the most predominant one in the Gulf region, Mediterranean region, and in Africa. However, travelers who seek medical care in different countries may bring back their endemic flora back to their home countries. So although we don't have KBC now circulating in our countries, we may see it one day, hopefully not. So let's make a quick comparison between Saudi Arabia and also in Kuwait versus the United States in terms of the epidemiology of carbapenemases. As you can see, KBC is the most predominant gene in the United States. However, this is very uncommon in the kingdom. In contrast, OXA48 is the most common in the kingdom and in the Gulf region, which is very uncommon in the United States. Before I move to pharmacological therapy, I would like to emphasize the importance of microbiological testing to guide the treatment approach. Once we know that this pathogen is carbapenem resistant, we can do what's called phenotypic testing to differentiate whether it's carbapenemase or not. And we can do also molecular testing or genetic testing to differentiate whether it's KBC, OXA48, or one of the metallo-beta-lactamases. What you see in the right lower corner is the gene expert CARBA-R that we have it in King Saud University Medical City, which can differentiate between the 5 different types of carbapenemase in 1 hour. This is a statement of the guideline that emphasizes the importance of having microbiological testing to treat multi-drug resistance. So how to treat CRE? Basically, those are the therapeutic options that we use to treat carbapenem-resistant Enterobacterales. Beta-lactam, mostly we are talking about novel beta-lactam, beta-lactamase inhibitor combinations. Ceftazidime avibactam, meropenem vaborbactam, imipenem rilibactam, cefdiericol, and aztreonam. On the other side, we have non-beta-lactams, the more traditional antibiotics, aminoglycoside, colistin, tigecycline, eravacycline. Those are the available options approved by the Saudi FDA, and we just got the approval for those 2 agents as well. Before the introduction of the novel beta-lactam, beta-lactamase inhibitor combinations, we were relying on more traditional therapies. By that time, we called them best available therapies. They could be monotherapy, dual therapy or triple therapy. They could be aminoglycoside based, they could be colistin based. You can imagine the risk of nephrotoxicity whenever we use both. They could be carbapenem based, they could be tigecycline based. And by that time, the clinicians selected whatever they want without a clear evidence in the literature. And after the approval of novel agents, I'm talking about the era after 2015, we realized how ineffective those agents were and how toxic they were as well. So, do those novel agents improve the clinical outcome compared to the standard of care or best available therapy by that time or more traditional therapies? The simple answer is absolutely yes. This is a summary of all the available literature and published in the last 4 years that compared the novel agents to more traditional therapeutics such as colistin. And as you can see, all of them showed reduced mortality compared to more traditional therapy. Beside the dramatic reduction in mortality, another important argument is the risk of nephrotoxicity. And as you can see, all of them, all of the novel agents, whether they, we are talking about ceftazidime avibactam or meropenem vaborbactam or imipenem relibactam, all of them reduce the nephrotoxicity compared to more traditional therapeutics. The rate of colistin nephrotoxicity in the literature is between 30 to 40%. So to summarize that, if we have the 3 different novel beta-lactam, beta-lactamase inhibitor combinations, which one is the most important for us in the Gulf region? If we want to compare all 3 of them, if we want to compare the 3 of them, so all of them can cover KBC. None of them covers New Delhi metallobeta-lactamase. However, they vary in terms of OXA48. So the only reliable one to be used for OXA48 is ceftazidime avibactam. This is why we need ceftazidime avibactam in our formularies in all the hospitals in the Gulf region to cover that important gene, which is very, very predominant in the Gulf region. So if we have KBC, we can use any of the 3 novel agents. If we have OXA48, we have only ceftazidime avibactam, other agent called cefdirichol as well, but we don't have it in the kingdom. And if we have metallobeta-lactamase, we have to use a combination between ceftazidime avibactam and aztreonam or cefdirichol alone. So let's talk about the first novel agent, ceftazidime avibactam. So the novelty of this agent comes from avibactam, the beta-lactamase inhibitor. So it expands the ceftazidime activity to include KBC, OXA48, non-carbapenemase producer as well. It retains activity against many metallobeta-lactamases, non-metallobeta-lactamases, and it's approved for UTI, intra-abdominal infection, and pneumonia. So we have very limited studies that compared the novel agents to more traditional therapy. One of them is the one that we published 2 years ago. And actually we conducted a multi-center study in the kingdom, and we recruited 230 patients in 5 different tertiary care hospitals to compare ceftazidime avibactam versus colistin, primarily against OXA48. And the result of our study showed that the novel agent ceftazidime avibactam improved the clinical success or clinical cure significantly, reduced the nephrotoxicity significantly, and it showed numerical reduction in mortality as well. And this is our study if you'd like to take a look. Another important study that was published in Italy, but it's very related to our epidemiology in the Gulf region, is the use of the combination ceftazidime avibactam plus aztreonam versus the more traditional therapy colistin to treat metallobeta-lactamase producing isolates. And simply the result showed that the use of this novel combination reduced the mortality significantly from 44% to 19%. And lastly, for ceftazidime avibactam, the same week we introduced this agent to our formulary in the hospital. We created a prescribing guide to restrict its use. We don't want it to be prescribed to everyone. So it should be specific for patients with isolates that are resistant to all traditional beta-lactams. So this is just a screenshot of the 3-page restricted criteria, and it's actually under the approval of the ID. And this is just a summary of the IDSA guideline for the treatment of CRE. Given that we have different epidemiology, so we don't apply everything in particular. We have to follow our epidemiology, our antibiograms, and give targeted therapeutics. However, this is just a summary of what they provided in 2013. As you can see, novel beta-lactam, beta-lactamase inhibitor combinations are the cornerstone for the treatment. However, we can use different agents for simple infections such as cystitis or pyelonephritis, such as fluoroquinolone or nitrofurantoin. So now if we have CRE pathogen, what should we do? So first of all, the treatment should be based on multiple factors: the susceptibility, carbapenemase gene if exists, and site of infection. If we have isolates susceptible to ceftazidime avibactam, we use it. If we have metallobeta-lactamase, which is resistant to everything, we use aztreonam plus ceftazidime avibactam or cefderichol if it's available. And if it's resistant to ceftazidime avibactam, we use more traditional therapeutics. Now I just finished. In the last 10 to 12 minutes, I will be talking about the drug-resistant Pseudomonas. So how prevalent drug-resistant Pseudomonas worldwide? So according to Sentry Antimicrobial Surveillance Program, which was established in 1997 and encompasses more than 750,000 isolates in 200 or 400 countries all over the world, the rate of multi-drug resistant Pseudomonas globally is 25%. So looking at the Gulf region in terms of epidemiology again, but here I will be focusing on carbapenem-resistant Pseudomonas. So the rate of carbapenem-resistant Pseudomonas in the kingdom is 30%. And again, we see lower rate in Kuwait, which is only 3%. So this is another study based on the GLASS program, which is a global antimicrobial resistance and use surveillance program, which is actually conducted by the WHO, showed that according to 8,000 isolates reported by the Kingdom of Saudi Arabia, the rate is 21% of MDR Pseudomonas, whereas it's only 3% in Kuwait. However, the number of isolates are different. So we had 8,000 isolates reported by the Kingdom with only 450 isolates reported from Kuwait. Again, this is the study we published a few years ago, and here we are focusing on multi-drug resistant Pseudomonas or carbapenem resistant Pseudomonas, and the rate reached to up to 15 to 20% again in our study. Here is the trend of antimicrobial resistance among Pseudomonas in the Kingdom of Saudi Arabia in the last 2 decades. So I believe that the understanding of the basic mechanism of resistance and how bacteria behave when exposed to antibiotics are crucial to understand the antimicrobial selection. The genetic versatility of Pseudomonas and its ability to co-regulate multiple resistance mechanisms, making Pseudomonas a constantly moving target and one of the greatest therapeutic challenge. We have 3 main resistance mechanisms for Pseudomonas for all traditional beta-lactam. All of them are considered chromosomally mediated. Loss of outer membrane purin, production of efflux pump, overexpression of MC. The reason why I explain them is that the novel agent ceftolozine and tazobactam acts by overcoming all these 3 resistance mechanisms. So those resistance mechanisms developed resistance against traditional beta-lactam, such as piperacillin tazobactam. However, when it comes to ceftolozane tazobactam, it becomes in vitro active. So it's FDA approved for the treatment of pneumonia, intra-abdominal infection, and UTI. However, a trick here is that we have to use double the dose for pneumonia as per the FDA approval. So the usual dose is 1.5 gram. However, when it comes to pneumonia, we have to double the dose simply because the blood to epithelial lining fluid penetration ratio is only 50%. So 50% of drug administered to the systemic circulation reach to the epithelial lining fluid. We also published a study about a patient with hemodynamic instability and with intermittent hemodialysis with HAP, and our pharmacokinetic study also suggested that we have to do double the dose for ceftolozane tazobactam to reach the same pharmacokinetic parameters for pneumonia, for intra-abdominal infection and UTI. So this agent is novel, as I mentioned, but let's talk here specifically about clinical studies. We have only 3 clinical studies until now that compared ceftolozane tazobactam to colistin. By that time, it was best available therapy to treat multi-drug resistant Pseudomonas, one of which is the one that we published 2 years ago. We recruited 184 patients in 5 different centers. And the study again showed significant improvement in clinical cure, significant reduction in nephrotoxicity, and also reduce mortality, but it was not statistically significant. This is the study if you'd like to take a look at it. So these are the 3 available studies nowadays. 2 of them were conducted in the Kingdom, 1 of them in the United States. All of them showed that ceftolozane tazobactam is associated with reduced mortality when compared to more traditional therapy, colistin. So the other agent that I just talked about for CRE is also useful for Pseudomonas because the avibactam component can inhibit one of the mechanisms of Pseudomonas resistance, which is the MC production. And it has the same FDA approval I just mentioned for ceftolozane tazobactam. Now the question is, we have 2 novel agents currently available in the Kingdom, Saudi FDA approved, and definitely it's available in Kuwait. And both of them can be used for multi-drug resistant Pseudomonas. Which one to use? So we have several studies that compared them in terms of in vitro activities. Some studies showed that one may be better than the other. Some studies showed that they are comparable. However, there is no single study to compare them clinically in terms of clinical outcome. Based on that, we published this study 6 months ago in antimicrobial agents and chemotherapy after comparing the 2 agents in terms of clinical outcome. And we recruited 200 patients from 6 centers in the Kingdom, and there was no statistical significant difference whatsoever in any of the primary or secondary outcomes, including the safety outcomes and nephrotoxicity. So we can use either. However, many clinicians still prefer ceftolozane tazobactam to reserve ceftazidime avibactam for CREs. The last concept is, do we need to use combination therapy to treat Pseudomonas? We don't have data on the novel agents. We have all data about more traditional therapeutics. But until now, we don't have a specific study that was designed to compare novel agents as a monotherapy, such as ceftolozane tazobactam alone, versus ceftolozane tazobactam plus colistin or aminoglycoside or tigecycline. And all what we have in the literature is just subgroup analysis from studies that were not designed for that purpose. So based on that, we conducted the study, not published yet, and we compared the use of monotherapy versus combination, and we recruited 200 patients as well. And the result of our study showed no statistical significant difference between mono versus combination in terms of clinical outcome. However, when we look to the acute kidney injury, we see 3-fold increase in nephrotoxicity when we use combination therapy. And this is, I would say, logical, given that aminoglycoside, colistin are very nephrotoxic. And the last slide is about, or last two slides are about the updates from the IDSA guideline for the management of multi or difficult to treat resistant Pseudomonas. And as you can see, the cornerstone for the treatment of difficult to treat resistant Pseudomonas is the use of novel agents, the combination between beta-lactam and beta-lactamase inhibitor combinations, and so on and so forth. Again, the same week we introduced this to our formulary. We created this prescribing guide to restrict its use. It should be restricted to save it for patients who need it in the future. So it should be only for patients who have Pseudomonas isolate resistant to all traditional beta-lactam. And the microbiology showed the in vitro susceptibility to cephalosporin tazobactam using e-test. And that's what I have for today. Thank you very much for listening.
Thank you so much for our speakers and all our attendees. I appreciate your time in coming here. So what do we have next? So in about half an hour, our poster presenters, or the winners of the best student poster and the winner of the best researcher poster, are going to present their poster presentation. And it's going to be a 15-minute presentation for each winner, and then we'll start the award ceremony. So please enjoy some refreshment and then come back again for the ceremony. Thank you so much.
Okay. So my name is Isra Ali, and I'm the Senior Research Assistant working in this grant. This grant, the PI is Prof. Williams Masucha, and the co-PI is Dr. Altaf Ramian, and the contributor, Ms. Aisha Al-Baluchi. The title of this grant is Dual COX-LOX Inhibition Prevents and Ameliorates Paclitaxel-Induced Mechanical Allodynia in Rats in a Cannabinoid Receptor-Dependent Manner. I know it's a very long title, so let's break in. So we'll have the normal introduction, objective message, results, and conclusion outline. Let's start. So paclitaxel is a chemotherapeutic agent. It is used for solid tumor, ovarian and lung cancer. Unfortunately, it causes painful neuropathy in 60 or 70% of the patient. This painful neuropathy does not have any recommended treatment by the FDA, either for prevention or treatment. The only thing that is available is duloxetine, but it's only recommended by the American Society of Clinical Oncology. There is no huge evidence to support the use of duloxetine. So we have an unmet medical need. I want to talk a little bit about the clinical picture of chemotherapy-induced neuropathy. It starts in the hands and the feet, as you can see here, and it has a range of negative symptoms and positive symptoms. The negative symptoms will be loss of sensations. The positive symptoms may be spontaneous pain, hyperalgesia. There are two terms that I want to discuss, is hyperalgesia and allodynia. Hyperalgesia is pain that you feel from something that normally provokes pain, but a person with peripheral neuropathy would feel pain more than the normal. Allodynia, however, is a stimulus that does not normally provoke pain, but for a person with a peripheral neuropathy, it will provoke pain. Like this quote, for example, the etching from it with the hand, it will cause pain for people with peripheral neuropathy. So here comes our drug. Lycophyllin is a substrate analog of arachidonic acid, and it blocks lipooxygenase and cyclooxygenase. And it has a safety profile better than non-steroidal anti-inflammatory. And we will now see why we chose lycophyllin. Because this is the arachidonic acid and endocannabinoid metabolism. The arachidonic acid is normally metabolized by cyclooxygenase to produce prostaglandin and by lipooxygenase to produce leukotrienes. Both of them are pro-inflammatory. In addition, the anandamide and 2-arachidonyl glycerol, both of them are endocannabinoid. Endocannabinoid, it means that they work on the cannabinoid receptor and it prevents pain. Cyclooxygenase and lipooxygenase enzyme are involved in their metabolism to some extent. So if lycophyllin blocks the activity of cyclooxygenase and lipooxygenase, that means less prostaglandin and less leukotrienes. And that means also higher of the endocannabinoid anandamide and 2-arachidonyl glycerol. So our hypothesis, we hypothesize that if we give paclitaxel along with lycophyllin, we can prevent or treat neuropathy. We will compare this effect of lycophyllin with that of indomethacin. It's a blocker of cyclooxygenase only, plus minocycline, it's a blocker of 5-lipooxygenase only. And we hypothesized also that this effect may be produced or mediated through cannabinoid receptor. So we have 3 objectives, and we will break them one by one. We will answer each question alone. For the first objective, does lycophyllin prevent neuropathic pain? So we have an established model of paclitaxel-induced mechanical allodynia. This established model, we give paclitaxel in 2 alternate days. Those are the green arrows. So we give paclitaxel in 2 alternative days. We measure the mechanical sensitivity on day 0, and we measure the mechanical sensitivity on day 9, and we always found mechanical allodynia on day 9. So for the prevention, we will give lycophyllin concomitantly with paclitaxel for 5 days at a dose of 50 milligram per kilogram, 1 hour before paclitaxel. And we will assess mechanical sensitivity at the end. How we are going to measure mechanical allodynia, we will use something that is called dynamic plantar stachiometer. And I hope the video will work. Let's see. I don't think it will work. No, it worked. Okay. So this is the one fray. It works by, there's a probe here, and this probe will touch the paw of the rat. When the rat, with an increased force, when the rat removes its paw, that means it felt pain. So if the number is higher, that means that the rat tolerated much pain. If the number is very less, like 10 or something, that means the rat did not tolerate much pain. So here we can see the results. First, you can find the timeline here. This is the baseline results, and this is day 9 post-paclitaxel. And here is the force that the rat withdrew its paw on. As you can find, paclitaxel, as expected, on day 9 produced allodynia. The force is 10 compared to the vehicle, which is above 20. When we gave lycophyllin concomitantly with paclitaxel, here paclitaxel induced allodynia, and lycophyllin reverted this allodynia to normal. So this means that lycophyllin can work for prevention. So the other objective we thought about, does it work for acute treatment? Can it alleviate already established allodynia? So for the purpose of this, we gave paclitaxel on 2 alternate days, as we do. And then on day 9, we assessed the mechanical sensitivity. We found that the mechanical allodynia already established, and then we gave lycophyllin. And we assessed the mechanical sensitivity for interval of 4 hours. So on day 9, paclitaxel induced the mechanical allodynia, and we gave 3 different doses of lycophyllin, 25 milligram, 50 milligram, and 100 milligram. This is a dose response curve because we wanted to choose the best dose for the next experiment. So actually all of the doses worked, like the 25, the 50, all of them reverted the allodynia to the normal level. Based on that graph, we chose the 50 milligram per kilogram because it's the dose in the middle. We did not want to go very high. And the last objective that we have is the effect of lycophyllin mediated through cannabinoid receptor or not. So, and we wanted also to compare it to endomycin plus minocycline, endomycin block cyclooxygenase, and minocycline block lipooxygenase 5. We gave also AM251, which is a cannabinoid receptor 1 antagonist, and AM630, which is a cannabinoid type 2 antagonist. So what did we expect? We expect if the effect is mediated through cannabinoid receptor, if we are blocking the receptor, then the effect will be inhibited. And that's what we actually found. So first, endomycin plus minocycline reverted the allodynia. Here, it's 30. And then AM251, which is a cannabinoid receptor type 1, blocked the effect, and AM60 did that as well. So that means that effect is through cannabinoid receptor. Here as well, you can find, because we compared endomycin and minocycline with lycophyllin, you can find that lycophyllin also was blocked by the effect of AM251 and AM60. That means that our effect of lycophyllin and minocycline plus endomycin could be mediated through cannabinoid receptor. So in summary, what we did, we give paclitaxel along with either lycophyllin or endomycin plus minocycline. We assessed the mechanical sensitivity, and we found that lycophyllin prevented and treated and paclitaxel induced mechanical allodynia in a cannabinoid receptor dependent manner. So in conclusion, this is a proof of concept experiment. What does that mean? That means that lycophyllin is the first in its class. So we can have others in the class that follow it. We can use other drugs that, like lycophyllin. It can open a wide array of experiments for us. Now, we are studying, what about the future recommendation? We are studying the gene and the protein that is involved in lycophyllin anti-allodenic effect. We are also studying the modulation of enzymatic activity of cyclooxygenase and lipooxygenase. In addition, we are going to measure the endocannabinoid levels in response to paclitaxel and lycophyllin administration to make sure that it's really mediated through the endocannabinoid receptor. So here is the poster. It's poster number 26. And here is the research team. Please join me to thank them all. This is Prof. Williams Masucha, the PI on this grant, Dr. Altaf, the co-PI in this grant, and Ms. Aisha Al-Balushi, my lab partner and teaching assistant, and me. So we all acknowledge the role of Kuwait University Research Sector for funding that project. And with that, I conclude my presentation. Thank you very much.
Now for the winner of the Best Student Poster Presentation, Maryam Al-Shimali. Please, Maryam.
Good afternoon. My name is Maryam Al-Shimali. Today I will present my project, Exploring the Arabian Gulf Macrofauna Diversity for the Discovery of a New Marine Leads, of a Novel Marine-Derived Leads. I will talk about the biodiversity of the marine environment, marine-derived drugs, marine macrofauna, sponges and tunicates, and I will talk about GMBS as a new web-based tool in the natural product discovery, objectives, methods, and the results of this project. Oceans represent the largest biosphere, which constitute more than 70% of Earth's surface, with a huge biodiversity and largely unexplored. Marine environment is considered a vital pipeline in supplying the drug discovery process with the novel natural products. Despite the biodiversity of the Kuwaiti marine environment, studies on the Kuwaiti marine organisms as a source of a new bioactive compounds is almost non-existent. The advancement of the marine natural product by the academia and the pharmaceutical industries have culminated into the introduction of several new marine-derived drugs to the clinic with different therapeutic properties, such as used in the oncology, infectious diseases, and metabolic diseases. Marine macrofauna, sponges. Sponges are a massive source of novel bioactive chemical compounds. From 2001 to 2010, approximately 30% of a new marine natural product were discovered from sponges, and moreover, from 2013 to 2019, several new marine natural products were discovered from the Dactyloceratida sponges. The second marine macrofauna is tunicates or ascidians. They are invertebrate animals, sessile, immobile, subtle, bound to hard surfaces or rocks. Researchers in the natural product field have investigated the pharmacological properties of these marine macrofauna in the development of a new drug candidates. to be effective in different human diseases. A new emerging method in the natural product research is the molecular networking, which is the fundamental key computational tool that establishes Global Natural Product Social Molecular Networking, GMBS. GMBS is an open access web-based interactive platform that aids in the isolation and the discovery of a new lead compounds through the visualization and the interpretation of the output of a mass spectral analysis. And the numerous of a natural product were identified and discovered by this approach. This tool relies on grouping and the arrangement of large data sets according to the spectral similarities between different but related precursor ion into groups, where each node representing a mass spectra and the edge connecting the two nodes indicating the mass similarities, which is calculated and assigned with a cosine score ranging from 0 to 1. And each group representing a molecular family. The objective of this project is to build up a marine-derived library, mainly from sponges and tourniquet collected from the Arabian Gulf Coast around Garo and al-Maradem Island. After that, extraction, fractionation, and the partitioning of these fractions and subjecting the library to both biological profiling and chemical profiling. After that, the chemical diversity of the collected samples will be further explored through the GMBS. The collection of marine samples, this is the Kuwait map and its 9 islands showing the collection site around al-Maradem and Garo Islands. 10 samples were collected on the average size of 5 grams. No hard corals or a rare organism were obtained, and the representative voucher specimen were cut and stored in a 70% aqueous solution and sent for taxonomic identification. Voucher samples, description sheets, photographs were sent for taxonomic identification and the Faculty of Science. The first organism was identified is Plagia nigra, black ascidians. The second organism was identified, Didymonis species, colonial ascidians, and the taxonomic identification was done by Prof. Huda Mahmoud, Department of Biological Sciences. After that, the extraction and the fractionation, only 7 samples presented in a good quantity and enough quantity to be further extracted. Each freeze-dried sample were cut, weighted, and subjected to non-polar extraction and the polar extraction using different solvent system, and the process were repeated 3 times. After that, the fractionation process involving solvent partitioning and silica gel chromatography. The library generation. In this library, each sample affords 2 main extracts, polar and non-polar, and 3 silica, 3 solvent partitions fractions, and 5 or 6 column fractions, resulting in the generation of 64 fractions with different polarities. The MTT profiling using the biological profiling using the MTT and the broth dilution assay. The heat map showing the percentage viability of the generated fraction against the MDA-MB231 cell breast cancer cells and Staphylococcus aureus gram-positive bacteria and the E. coli gram-negative bacteria. We show that some fractions showed a positive activity against the MDA-MB231 cells. However, no antibacterial effect were observed. The cells and the bacterial culture were treated with the generated fractions at 3 different concentrations. After that, the active ethyl acetate fractions were subfractionated over the silica gel chromatography and tested against the MDA-MB231 cells, breast cancer cells. After the biological profiling and the NMR analysis, the sample were prioritized for further purification, and they were prioritized, and their LC-MS-MS was done, and the HPLC profiling was done, and were analyzed by GMBS to create the molecular network. This is the molecular network of selected prioritized marine fractions from 6 marine samples. The molecular network showing some identified compounds when compared to the library. However, some compounds were unidentified. And here we are looking at the sub-molecular families and the sub-networks. They were annotated in 5 different marine species, and these 14 nodes are unidentified compound which will be further prioritized for future HPLC purification, undergoing the discovery of a new therapeutically active marine-derived leads. So in conclusion, Kuwaiti marine tunicates mainly collected from the Arabian Gulf Coast around Garo and Al Maradif Island. A library was generated of nearly 70 extracts and fractions, and the library were subjected to and screened against MDA MB231 cells, breast cancer cells, and 2 strains of gram-positive and gram-negative bacteria. The overall profiling addressed the crucial role of marine natural product to be potentially for the potential development of anti-tumor agent. However, no antibacterial effect were showed in this study. After that, the sample were analyzed by GMBS that showed some molecular networks that potentially contain some uniquely molecule to be further identified in the future. To our knowledge, this is the first study of its kind in Kuwait, aiming for the discovery of a new marine natural product in Kuwait. By the end, I want to thank my supervisor, Prof. Khalid Arabi, and my co-supervisor, Dr. Fatma Al-Awadi. Special thanks to Prof. Huda Mahmoud, her assistant in the taxonomic identification. This project is financially supported by College of Graduate Study and Research Sector. Special thanks to the RCF and HSC and the RSPU and the Faculty of Science for their aids in the mass analysis. Thank you.
Thank you, Mariam, so much for your presentation. In about 5-10 minutes, we're going to start the closure remarks and also the award ceremony. We're just awaiting our last speaker, and then we will be done. Please wait for us.
Bismillah ar-Rahman ar-Rahim. As we are closing the conference, we would like to thank all people who attended this 8th Kuwait International Pharmacy Conference, the KIPC 2024. We would like to thank you all for coming, and we hope you benefit. And inshallah, we'll see you next in 2 years. Yes. Now we would like to thank the speakers. Professor Abdu Adam, Department of Pharmacology, Faculty of Medicine and Health Science, Khalifa University, United Arab Emirates. Dr. Thamer Al-Mangour, Licensed Consultant Pharmacist, Saudi Arabia, Commission for Health Specialists, Saudi Arabia, Kingdom of Saudi Arabia. Dr. Ahmed Al-Demirdash, Department of Clinical Pharmacy, College of Pharmacy, King Saud University, Kingdom of Saudi Arabia. Professor Khalid Urabi, Department of Pharmaceutical Chemistry, College of Pharmacy, Kuwait University. Dr. Nada Al-Hasawi, Department of Pharmaceutical Chemistry, College of Pharmacy, Kuwait University. Dr. Abdul Qadir Mutwalli, Department of Pharmaceutics, College of Pharmacy, Kuwait University. And Dr. Abdurrahman Al-Shaya from King Saud University. Dr. Fatma Al-Rashid from the Department of Pharmacy Practice, College of Pharmacy, Kuwait University, and also the chairperson of the KIPC 2024. Professor Ahmed Ous from the Department of Pharmacology and Therapeutics, College of Pharmacy, Kuwait University. And now we go with the poster award winners. Best Researcher, Isra Ali. Again, Best Researcher, Ms. Rania Muhammad Al-Anwar. Also for Best Researcher, Ms. Noor As-Safar. Now we go for the Best Student Researcher, Pharmacist Maryam Al-Shimali. Again, with Best Student Researcher, Mr. Abdullah Nader. Best Student Researcher, Ms. Shog Al-Azmi. Thank you all for attending. We hope you enjoyed the conference and benefit from it. Thank you very much. Enjoy the rest of the evening.